Within you lies a landscape more intricate than any mountain range, more mysterious than the deepest ocean trench. This is your digestive system, a 30-foot odyssey that transforms the outside world into the very substance of your being. Every morsel you taste embarks upon an epic voyage through caverns that crush and chambers that churn, through passages lined with millions of tiny gates that select what becomes you and what passes through. Here, in the gentle darkness of your interior, chemistry becomes alchemy. A simple apple becomes amino acids that will repair your muscles, glucose that will fuel your thoughts, and vitamins that will brighten your vision. This is the story of transformation itself, told in the quiet rhythm of peristalsis and the patient work of enzymes, where every meal writes another chapter in the ongoing narrative of your existence. In the beginning, there is the mouth. This remarkable cavern serves as the gateway to your internal universe, where the first notes of digestion's grand symphony are played. Here, in this warm, moist chamber, the transformation begins not with violence, but with precision, not with haste, but with careful orchestration of mechanical and chemical forces that have been perfected over millions of years of evolution. The mouth is far more than a simple opening. It is an architectural marvel, a complex ecosystem where 32 precisely shaped tools work in harmony with 3 pairs of salivary glands, a muscular tongue, and a sophisticated sensory network that can detect the subtlest changes in texture, temperature, and chemical composition. This is where your body first encounters the outside world in its most intimate way, where foreign matter begins its metamorphosis into the very substance of your being. Consider the teeth, those remarkable structures that many take for granted. Each tooth in your mouth is a specialized instrument shaped by evolution for a specific purpose. The sharp chisel-like incisors at the front slice through food with surgical precision, their edges maintained by constant use and the natural abrasion that keeps them keen. Behind them stand the canines, pointed spears designed for tearing, remnants of our more carnivorous past, yet still essential for breaking through tougher textures. The premolars and molars that line the sides of your mouth are perhaps the most impressive of all. These broad, ridged surfaces work like ancient millstones, grinding and crushing with a force that can exceed 200 pounds per square inch. The complex topography of their crowns, with valleys called fissures and peaks called cusps, creates a landscape perfectly designed for reducing food to smaller and smaller fragments. This is not random destruction, but calculated deconstruction, breaking down complex structures into forms that can be more easily handled by the chemical processes that follow. Yet mastication is more than mere mechanical breakdown. Each chew releases volatile compounds that rise through the back of the mouth to the olfactory receptors in the nose, contributing to what we perceive as flavor. The longer you chew, the more complete this release becomes, which is why your grandmother's advice to chew slowly had scientific merit. The mechanical action also increases the surface area of food exponentially, creating more opportunities for enzymatic action to take place. As your teeth perform their careful demolition, your salivary glands respond with remarkable precision. 3 pairs of major glands work in concert with hundreds of minor glands scattered throughout the oral cavity. The parotid glands, the largest, sit just in front of your ears and produce a thin, enzyme-rich saliva that flows through Stenson's ducts. The submandibular glands beneath your jaw contribute a mixed secretion that is both lubricating and enzymatically active. The sublingual glands under your tongue add their own contribution, creating a complex mixture that is perfectly suited to begin the digestive process. Saliva itself is a marvel of biological engineering. This clear fluid, which you produce roughly 1 to 2 liters of each day, is far more complex than water. It contains electrolytes that maintain the proper pH, mucins that lubricate and protect, antibodies that defend against pathogens, and most importantly for digestion, enzymes that begin the chemical breakdown of food. The most significant of these enzymes is alpha amylase, also known as pyelin. This remarkable protein begins the breakdown of starches immediately upon contact, cleaving the long chains of glucose molecules that make up complex carbohydrates. Within moments of taking a bite of bread or potato, amylase is already at work, converting starch into smaller sugars. If you've ever noticed bread becoming slightly sweet the longer you chew it, you've experienced amylase in action. But saliva's role extends far beyond enzymatic activity. Its alkaline pH, typically ranging from 7 to 7.4, begins the crucial process of buffering against the acid that awaits in the stomach. The bicarbonate ions in saliva neutralize acids produced by bacteria in the mouth, protecting tooth enamel and maintaining the proper environment for enzymatic function. Without this constant buffering action, the delicate balance of the oral environment would quickly deteriorate. The production of saliva is controlled by both the sympathetic and parasympathetic nervous systems, responding not just to the presence of food, but to the mere thought of it. The sight of an appetizing meal, the aroma of cooking food, even the memory of a favorite dish can trigger salivary flow. This anticipatory response demonstrates the remarkable integration between your nervous system and digestive function, preparing your body for the work ahead even before the first bite is taken. The tongue, that muscular marvel that fills much of the oral cavity, plays multiple roles in this opening act of digestion. Its surface is covered with thousands of taste buds, each containing clusters of specialized chemoreceptors that can detect sweet, sour, salty, bitter, and umami flavors. But the tongue is far more than a sensory organ. Its powerful muscles manipulate food during chewing, positioning it between the teeth for optimal grinding, then collecting the chewed mass and forming it into a bolus suitable for swallowing. The formation of the bolus is a critical step that often goes unnoticed. The tongue works with remarkable precision, gathering the chewed food particles and mixing them with saliva to create a cohesive mass. The consistency of this bolus must be just right, not too dry or it cannot be swallowed safely, not too wet or it will fall apart during transport. The tongue assesses this consistency through mechanoreceptors embedded in its surface, making real-time adjustments to ensure successful swallowing. Temperature regulation also begins in the mouth. Hot foods must be cooled to prevent damage to the delicate tissues of the esophagus and stomach, while cold foods may need warming to optimize enzymatic activity. The mouth accomplishes this through its rich blood supply and the mixing action of the tongue, which spreads food over the warm surfaces of the oral cavity. As the bolus nears completion, the process of swallowing prepares to begin. This transition marks a crucial moment in the digestive journey, where voluntary control gives way to involuntary reflexes, where the conscious act of eating transforms into the automatic processes of internal transport. The tongue positions the bolus at the back of the mouth, triggering sensory receptors that will initiate one of the most complex reflexes in the human body. The oral phase of digestion, though brief, sets the stage for everything that follows. The mechanical breakdown accomplished by the teeth, the enzymatic action initiated by amylase, the formation of the bolus, and the sensory evaluation of the food all contribute to optimizing the efficiency of subsequent digestive processes. The mouth serves as both gatekeeper and processor, determining not just what enters the body, but how well prepared it is for the journey ahead. This initial processing is so important that problems in the oral phase can cascade through the entire digestive system. Poor chewing leads to larger food particles that are more difficult for the stomach to process. Inadequate saliva production can compromise both mechanical and chemical breakdown, while problems with bolus formation can lead to swallowing difficulties that may affect nutrition and health. As the bolus sits poised at the back of the tongue, ready for its descent into the esophagus, the mouth has completed its first crucial task. The food that entered as a complex mixture of proteins, carbohydrates, fats, vitamins, and minerals has been mechanically reduced, chemically modified, and carefully prepared for the next stage of its transformation. The symphony of digestion has begun, and now the bolus must travel through the muscular highway that will carry it to the stomach's processing chambers. As the carefully prepared bolus sits at the back of the tongue, poised for its journey deeper into the body, it stands at the threshold of one of the most remarkable transport systems in human anatomy. The esophagus, a muscular tube measuring approximately 10 inches in length, serves as the crucial highway connecting the mouth to the stomach. This seemingly simple conduit is actually a sophisticated piece of biological engineering capable of moving food against gravity through coordinated waves of muscular contraction while simultaneously protecting the delicate respiratory passages from contamination. The moment of swallowing marks a profound transition in the digestive process. What begins as a conscious, voluntary act quickly transforms into an involuntary reflex controlled by the brainstem. This transition occurs when the bolus touches specialized sensory receptors at the back of the tongue and in the pharynx, triggering what physiologists call the swallowing reflex or deglutition. In that instant, conscious control gives way to an ancient, automatic program that has been refined over millions of years of evolution. The swallowing process unfolds in three distinct phases, each precisely timed and coordinated. The oral phase, which we control voluntarily, involves the tongue pushing the bolus backward toward the pharynx. The pharyngeal phase, lasting about 1 second, is entirely involuntary and involves a complex choreography of muscle contractions and airway protection. Finally, the esophageal phase carries the bolus through the esophageal tube to the stomach, a journey that typically takes between 6 and 10 seconds, depending on the consistency of the swallowed material. During the pharyngeal phase, the body performs a remarkable feat of coordination that prevents food from entering the respiratory system. The soft palate elevates to seal off the nasal passages, while the larynx rises and the epiglottis folds down to cover the opening to the trachea. The vocal cords also close tightly, creating multiple barriers between the swallowed material and the lungs. Meanwhile, the upper esophageal sphincter, a ring of muscle at the top of the esophagus, relaxes to allow the bolus to enter the esophageal tube. The esophagus itself begins just below the level of the sixth cervical vertebra and descends through the neck and chest before passing through the diaphragm to connect with the stomach. This journey takes the esophagus through three distinct anatomical regions: the cervical esophagus in the neck, the thoracic esophagus in the chest cavity, and the short abdominal portion below the diaphragm. Each region presents unique challenges and characteristics that influence how food moves through this vital conduit. The wall of the esophagus consists of 4 distinct layers, each serving specific functions in the transport process. The innermost mucosa is lined with stratified squamous epithelium, the same type of tissue that lines the mouth and throat. This tough, protective lining can withstand the mechanical abrasion of passing food while remaining flexible enough to expand and contract with each swallow. Beneath the mucosa lies the submucosa, containing blood vessels, lymphatics, and mucus secreting glands that help lubricate the passage. The third layer, the muscularis propria, is perhaps the most remarkable feature of the esophagus. This muscular layer has a unique composition that reflects the esophagus's position between voluntary and involuntary control. The upper third consists primarily of skeletal muscle under direct control of the central nervous system. The middle third contains a mixture of skeletal and smooth muscle representing a transition zone. The lower third is composed entirely of smooth muscle, controlled by the autonomic nervous system and capable of the sustained rhythmic contractions necessary for efficient transport. The outermost layer, the adventitia, consists of connective tissue that anchors the esophagus to surrounding structures while allowing the flexibility necessary for the constant motion of swallowing and breathing. This layer also contains the nerve networks that coordinate esophageal function, including branches of the vagus nerve that carry signals between the brain and the digestive system. The process that moves food through the esophagus is called peristalsis, from the Greek words meaning to clasp and compress. This wave-like pattern of muscular contraction begins at the moment the bolus enters the esophagus and progresses systematically down its length. The coordinated contraction is so precise that food can travel from mouth to stomach even when a person is hanging upside down, demonstrating that this transport system relies not on gravity but on active muscular propulsion. Primary peristalsis begins with the swallowing reflex itself, as the upper esophageal sphincter relaxes to admit the bolus. A ring of muscular contraction begins just above the food mass. This ring of contraction, typically measuring about 4 centimeters in length, progresses down the esophagus at a speed of approximately 3 to 4 centimeters per second, pushing the bolus ahead of it like a piston in a cylinder. The pressure generated by this peristaltic wave is considerable, often exceeding 100 millimeters of mercury. This force is necessary to overcome not only the resistance of the esophageal walls, but also the negative pressure in the thoracic cavity that tends to collapse the esophagus. The wave must also be strong enough to open the lower esophageal sphincter, a ring of muscle that guards the entrance to the stomach and normally maintains a pressure of about 20 millimeters of mercury to prevent stomach contents from refluxing back into the esophagus. If the initial peristaltic wave fails to clear the esophagus completely, secondary peristalsis can be triggered by the continued presence of food in the esophageal lumen. This secondary wave is initiated by local stretch receptors in the esophageal wall and does not require another swallow to initiate. This backup mechanism ensures that even sticky or poorly formed boluses are eventually transported to the stomach. The lower esophageal sphincter represents one of the most important barriers in the digestive system. This physiological valve, located where the esophagus joins the stomach, must perform a delicate balancing act. It must remain tightly closed most of the time to prevent the highly acidic stomach contents from refluxing into the esophagus, yet it must relax completely and immediately when a peristaltic wave approaches. The sphincter receives dual innervation from both excitatory and inhibitory nerve fibers, allowing precise control of its function. The relaxation of the lower esophageal sphincter is coordinated with the approaching peristaltic wave through a phenomenon called receptive relaxation. As soon as swallowing begins, nerve signals travel ahead of the peristaltic wave to prepare the sphincter for opening. This anticipatory relaxation ensures that the sphincter is already open by the time the bolus arrives, preventing the backup that would occur if the wave reached a closed valve. The esophagus must also deal with the unique challenge of passing through the diaphragm, the dome-shaped muscle that separates the chest cavity from the abdominal cavity. The esophageal hiatus, the opening in the diaphragm through which the esophagus passes, is reinforced by muscular fibers that help support the lower esophageal sphincter. These crural fibers contribute to the anti-reflux mechanism by providing additional compression when abdominal pressure increases, such as during coughing or straining. Temperature regulation continues to be important during esophageal transit. Extremely hot or cold foods can trigger esophageal spasm, disrupting normal peristalsis. The esophagus has a rich blood supply that helps moderate temperature extremes, but severe thermal stress can still interfere with normal function. This is why extremely hot beverages can cause discomfort and why ice-cold drinks sometimes seem to stick temporarily during swallowing. The neural control of esophageal function is remarkably complex, involving both central and peripheral nervous systems. The swallowing center in the brainstem coordinates the initial phases of swallowing, while the intrinsic nervous system of the esophagus, called the myenteric plexus, controls the progression of peristaltic waves. The vagus nerve provides the primary neural pathway between the brain and the esophagus, carrying both motor commands and sensory information. Dysfunction of the esophagus can have profound effects on nutrition and quality of life. Achalasia, a condition in which the lower esophageal sphincter fails to relax properly, can make swallowing extremely difficult. Gastroesophageal reflux disease occurs when the sphincter is too weak, allowing stomach acid to damage the esophageal lining. Esophageal spasm can disrupt normal peristalsis, causing food to become trapped in the esophageal tube. The esophagus also serves as an important barrier to infection. Its stratified squamous epithelium provides protection against pathogens, while the constant flow of saliva and the mechanical action of peristalsis help clear bacteria and other microorganisms. The esophageal mucosa produces antimicrobial substances and contains immune cells that can respond to potential threats. As the bolus completes its journey through the esophagus and approaches the lower esophageal sphincter, it has been transformed from the relatively dry, mechanically processed food mass that left the mouth into a well-lubricated, cohesive bolus ready for the next phase of digestion. The muscular work of the esophagus has delivered the food safely and efficiently to the stomach while protecting the respiratory system and maintaining the integrity of the digestive tract. The transition from esophagus to stomach marks another crucial milestone in the digestive journey. The bolus that began as conscious bites taken in the mouth has now traveled through the body's most sophisticated transport system, protected by multiple sphincters and barriers, propelled by coordinated waves of muscular contraction and delivered precisely to the stomach's entrance. As the lower esophageal sphincter opens to admit this cargo, the bolus prepares to enter a very different environment, one where mechanical processing gives way to chemical assault, where the gentle pH of saliva will be overwhelmed by powerful acids, and where the journey of transformation will continue with even greater intensity. The stomach awaits like a patient chemist, prepared to perform one of the most remarkable transformations in the human body. As the bolus passes through the lower esophageal sphincter, it enters an environment so hostile that few organisms can survive it, yet so precisely controlled that it never harms its own walls. This muscular sac, roughly the size of a large grapefruit when empty, possesses the extraordinary ability to expand to accommodate up to 1.5 liters of food and liquid, while simultaneously producing acid strong enough to dissolve metal and enzymes capable of breaking down the most complex proteins. The stomach's anatomy reflects its dual nature as both storage vessel and processing chamber. Shaped somewhat like a curved flask, it consists of four distinct anatomical regions, each specialized for specific functions in the digestive process. The cardia, the narrow entrance where the esophagus connects, serves as the receiving area for incoming food. The fundus, the dome-shaped upper portion, acts primarily as a storage area capable of remarkable expansion without significant increase in pressure. The body, the largest section, serves as the main mixing and processing chamber. Finally, the antrum, the narrow lower section, functions as a grinding mill where food is reduced to the proper consistency for passage into the small intestine. The stomach wall itself is a masterpiece of biological engineering, composed of four distinct layers that work in harmony to accomplish the organ's complex functions. The innermost mucosa contains specialized cells that produce the stomach's powerful secretions while protecting themselves from destruction. This layer is arranged in deep folds called rugae that allow for dramatic expansion as food enters the stomach. The submucosa contains the blood vessels and nerves that support gastric function, while the muscularis propria consists of three layers of smooth muscle arranged in different orientations to provide the complex mixing and grinding motions necessary for mechanical digestion. The gastric mucosa contains several types of highly specialized cells, each contributing essential components to the digestive process. The surface mucus cells produce a thick, alkaline mucus that forms a protective barrier against the stomach's own acid. This mucus layer is so effective that the pH at the stomach's surface remains nearly neutral despite the presence of acid, with a pH below 2 just millimeters away. Parietal cells, found primarily in the fundus and body of the stomach, produce both hydrochloric acid and intrinsic factor, a protein essential for vitamin B12 absorption. Chief cells secrete pepsinogen, the inactive precursor of the powerful protein-digesting enzyme pepsin. The production of gastric acid represents one of the most energetically expensive processes in the human body. Parietal cells use specialized structures called canaliculi to concentrate hydrogen ions and transport them into the stomach lumen. This process requires enormous amounts of energy, with parietal cells containing more mitochondria per cell than almost any other cell type in the body. The hydrogen ions are combined with chloride ions to form hydrochloric acid, creating a solution so acidic that it would quickly destroy most biological tissues. The stomach's acid production is regulated by a sophisticated system of neural, hormonal, and local controls that ensure acid is produced only when needed and in appropriate quantities. The sight, smell, or even thought of food triggers the cephalic phase of gastric secretion mediated by the vagus nerve. This preparatory response can account for up to 30% of total acid production, demonstrating the intimate connection between the nervous system and digestive function. The gastric phase begins when food actually enters the stomach, triggering both mechanical and chemical sensors that stimulate further acid production. The intestinal phase provides feedback from the small intestine, modulating gastric secretion based on the rate of gastric emptying and the composition of the food being processed. Pepsinogen, the inactive enzyme precursor secreted by chief cells, undergoes a remarkable transformation in the acidic environment of the stomach. The low pH causes pepsinogen to spontaneously convert to its active form, pepsin, one of the most powerful protein digesting enzymes in the human body. Pepsin works optimally in the highly acidic environment of the stomach, cleaving proteins at specific amino acid sequences to produce smaller fragments called polypeptides. This enzymatic action works in concert with the stomach's mechanical churning to break down even the most complex protein structures. The mechanical aspects of gastric digestion are equally impressive. The stomach's three-layered muscle wall creates a pattern of contractions that serves multiple functions. Weak contractions in the fundus help mix newly arrived food with gastric secretions, while stronger waves that begin in the body of the stomach propel food toward the antrum. The most powerful contractions occur in the antrum, where food is subjected to intense grinding action against the closed pyloric sphincter. This process, called trituration, reduces food particles to a uniform size of approximately 1 to 2 millimeters, the optimal size for efficient digestion in the small intestine. The pyloric sphincter, a ring of smooth muscle at the junction between the stomach and duodenum, serves as a sophisticated filter that controls the rate at which processed food leaves the stomach. This valve opens in response to specific mechanical and chemical signals, allowing properly processed food to pass while retaining material that requires further processing. The sphincter remains closed most of the time, opening only when approached by appropriately sized particles of the right consistency and chemical composition. Gastric emptying is a carefully regulated process that depends on numerous factors, including the volume, composition, and temperature of the stomach contents. Liquids typically empty more rapidly than solids, with water leaving the stomach within 30 minutes, while solid meals may require 2 to 4 hours for complete emptying. Fats slow gastric emptying more than any other macronutrient, which explains why fatty meals tend to promote satiety and why they can sometimes cause feelings of fullness or discomfort. The stomach's protective mechanisms are as important as its digestive functions. The mucus barrier produced by surface cells forms the first line of defense against acid damage. This barrier is maintained by prostaglandins, hormone-like substances that stimulate mucus production and promote the rapid replacement of damaged surface cells. The stomach lining renews itself completely every 3 to 5 days, one of the fastest regeneration rates in the human body. This rapid turnover is essential given the harsh chemical environment in which these cells must function. Blood flow to the stomach increases dramatically during digestion, delivering the oxygen and nutrients necessary for increased cellular activity, while also helping to buffer against potential acid damage. The stomach's rich vascular supply also facilitates the rapid transport of absorbed substances, though the stomach itself absorbs relatively few nutrients compared to other portions of the digestive tract. Water, alcohol, and some medications can be absorbed directly through the gastric mucosa, but most nutritional absorption occurs in the small intestine. The stomach also produces several important hormones that coordinate digestive function. Gastrin, produced by G cells in the antrum, stimulates acid production and promotes gastric motility. Ghrelin, often called the hunger hormone, is produced primarily by the stomach and signals the brain when it's time to eat. Somatostatin, produced by D cells throughout the digestive tract, acts as a brake on gastric secretion when acid levels become too high or when the small intestine signals that it cannot handle more material. The transformation that occurs in the stomach is both physical and chemical. The solid food that entered as a bolus is converted into a semi liquid mixture called chyme, with a consistency similar to thick soup. This chyme contains partially digested proteins, emulsified fats, and dissolved carbohydrates, all suspended in the acidic gastric secretions. The acid environment not only activates pepsin, but also helps solubilize minerals like iron and calcium, making them available for absorption in the small intestine. Temperature regulation continues to be important in the stomach. Very hot or cold foods can disrupt normal gastric function, while moderately warm foods promote optimal enzyme activity and motility. The stomach's blood supply helps moderate temperature extremes, gradually bringing very hot or cold items closer to body temperature. The stomach's ability to accommodate varying volumes of food while maintaining appropriate processing conditions is truly remarkable. This adaptive capacity is mediated by both neural and local mechanisms that allow the stomach wall to relax as food enters, preventing excessive increases in pressure that could interfere with digestion or cause discomfort. This accommodation also ensures that gastric acid remains properly concentrated despite dilution by food and beverages. Certain substances can interfere with normal gastric function. Alcohol can directly damage the gastric mucosa while simultaneously stimulating acid production, creating conditions that favor ulcer formation. Nonsteroidal anti-inflammatory drugs, NSAIDs, can suppress the prostaglandins that maintain the protective mucus barrier, leaving the stomach vulnerable to acid damage. Helicobacter pylori bacteria can colonize the stomach despite its acidic environment, leading to chronic inflammation and increased risk of ulcers and stomach cancer. The stomach's role extends beyond simple digestion to include important defensive functions. Its highly acidic environment destroys most bacteria, viruses, and other pathogens that might be present in food, serving as a crucial barrier against foodborne illness. This sterilizing effect is so powerful that very few microorganisms can survive passage through a healthy stomach. As the chyme nears readiness for passage into the small intestine, it has been transformed from recognizable food into a homogeneous mixture optimized for the next phase of digestion. Proteins have been partially broken down into polypeptides, fats have been emulsified into smaller droplets, and the overall pH has been adjusted to activate the pancreatic enzymes that await in the duodenum. The mechanical processing has reduced particle size to ensure efficient contact with intestinal enzymes, while the chemical processing has begun the complex task of liberating nutrients from their original food matrices. The journey through the stomach represents a crucial transition in the digestive process. What entered as a semi-solid bolus exits as liquid chyme, transformed by one of the most powerful chemical environments in the human body, yet delivered with remarkable precision to the next stage of processing. As the pyloric sphincter opens to release small portions of chyme into the duodenum, the stomach continues its rhythmic contractions, ensuring that each portion of food receives adequate processing before moving on to the absorption-focused environment of the small intestine. Hidden deep within the abdominal cavity, Nestled in the curve of the duodenum, like a fish resting in the bend of a river, lies one of the most industrious organs in the human body. The pancreas, measuring roughly 6 inches in length and weighing about 100 grams, serves as both a hormonal control center and a biochemical factory of extraordinary sophistication. This elongated, somewhat flattened organ performs a dual role that makes it unique among digestive organs. It functions simultaneously as an endocrine gland, producing hormones that regulate blood sugar, and as an exocrine gland, manufacturing the most powerful digestive enzymes in the human body. The pancreas can be divided into 4 distinct anatomical regions, each contributing to its overall function. The head, the widest portion, nestles snugly against the C-shaped curve of the duodenum and contains the main pancreatic duct's exit point. The neck, a narrow transitional area, connects the head to the body, the longest central portion of the organ. Finally, the tail extends toward the spleen, tapering to a point that nearly touches this important lymphatic organ. This anatomical arrangement places the pancreas in an ideal position to monitor the chemical composition of newly arrived chyme and respond with precisely calibrated secretions. The exocrine pancreas, which comprises about 85% of the organ's mass, consists of thousands of small, grape-like clusters called acini. Each acinus contains specialized cells called acinar cells, which are among the most metabolically active cells in the human body. These remarkable cells contain an abundance of rough endoplasmic reticulum, giving them a distinctive basophilic appearance under the microscope and reflecting their primary function as protein factories. The acinar cells manufacture and store the precursors of digestive enzymes, maintaining a constant readiness to respond to the arrival of food in the duodenum. The pancreatic juice produced by these acinar cells is a clear, colorless fluid with a pH ranging from 8 to 8.3, making it strongly alkaline. This alkalinity is crucial for neutralizing the highly acidic chyme that arrives from the stomach. The stomach's acid, with a pH as low as 1.5, must be buffered to create the optimal environment for pancreatic enzyme function. The bicarbonate ions in pancreatic juice accomplish this neutralization, raising the pH of the duodenal contents to approximately 7.5 to 8.5, the range in which pancreatic enzymes work most efficiently. The volume of pancreatic juice produced daily is impressive, ranging from 1.2 to 1.5 liters in a healthy adult. This substantial output reflects the pancreas's critical role in digestion, as virtually all enzymatic breakdown of proteins, fats, and complex carbohydrates depends on pancreatic secretions. The composition of this juice changes dynamically based on the body's needs, with hormone-sensitive cells adjusting both the volume and enzyme concentration in response to the type and amount of food being processed. The pancreatic enzymes themselves represent one of nature's most sophisticated biochemical arsenals. These proteins are initially synthesized and stored as inactive precursors called zymogens, a safety mechanism that prevents the pancreas from digesting itself. The activation of these enzymes occurs only after they reach the duodenum, where specific conditions trigger their conversion to active forms. Trypsinogen, the most important of these precursors, is converted to trypsin by enterokinase, an enzyme produced by the duodenal mucosa. This conversion represents a critical control point in pancreatic enzyme activation, as trypsin then catalyzes the activation of other pancreatic enzymes, including chymotrypsinogen to chymotrypsin, proelastase to elastase, and procarboxypeptidases to carboxypeptidases. This cascade system ensures that enzyme activation occurs only in the appropriate location and prevents potentially dangerous enzyme activity within the pancreas itself. Trypsin and chymotrypsin are both endopeptidases, meaning they cleave proteins at specific internal amino acid sequences rather than removing amino acids from the ends of protein chains. Trypsin specifically targets proteins at lysine and arginine residues, while chymotrypsin prefers to cut at phenylalanine, tryptophan, and tyrosine residues. This complementary activity ensures comprehensive breakdown of dietary proteins into smaller peptide fragments that can be further processed by other enzymes. Elastase, another endopeptidase, specializes in breaking down elastin and other fibrous proteins that give structure to meat and plant tissues. Carboxypeptidases, A and B, are exopeptidases that remove amino acids from the carboxyl end of peptide chains, with carboxypeptidase A preferring aromatic and branched amino acids, while carboxypeptidase B targets basic amino acids. Together, these protein digesting enzymes can reduce even the most complex dietary proteins to small peptides and individual amino acids. Pancreatic lipase represents the primary fat digesting enzyme in the human digestive system, capable of breaking down triglycerides into monoglycerides and fatty acids. This enzyme works in concert with colipase, a cofactor that helps lipase bind to fat droplets in the presence of bile salts. The activity of pancreatic lipase is so efficient that it can digest up to 90% of dietary fats, making it essential for the absorption of fat-soluble vitamins and the utilization of dietary lipids. Phospholipase, A2, is another important lipolytic enzyme that specifically targets phospholipids, complex lipids that are major components of cell membranes. This enzyme cleaves fatty acids from the second carbon of phospholipids, producing lysophospholipids and free fatty acids. Cholesterol esterase breaks down cholesterol esters, freeing cholesterol for absorption and further metabolism. The pancreatic amylase continues the work begun by salivary amylase, breaking down complex carbohydrates into smaller oligosaccharides. This enzyme is particularly important for processing starches that were not completely digested in the mouth, as well as starches that were protected from salivary amylase by the food matrix. Pancreatic amylase works optimally in the alkaline environment of the duodenum and can completely convert starch to maltose and other small oligosaccharides. The regulation of pancreatic secretion involves a sophisticated interplay of neural, hormonal, and local factors that ensure enzymes are released in appropriate quantities and at optimal timing. The cephalic phase of pancreatic secretion begins even before food enters the stomach, triggered by the sight, smell, or thought of food. The vagus nerve stimulates both acinar cells and ductal cells, preparing the pancreas for the digestive work ahead. The gastric phase of pancreatic secretion is relatively modest, primarily mediated by vagal stimulation as the stomach begins processing food. However, the intestinal phase represents the major stimulus for pancreatic enzyme release. As acidic chyme enters the duodenum, specialized cells in the duodenal mucosa release hormones that coordinate pancreatic function with the body's digestive needs. Cholecystokinins, often abbreviated as CCK, is perhaps the most important hormone regulating pancreatic enzyme secretion. Released by eye cells in the duodenal mucosa, in response to the presence of fats and proteins. CCK stimulates the release of enzyme rich pancreatic juice, while simultaneously causing the gallbladder to contract and release bile. This coordinated response ensures that fat digestion proceeds efficiently, with both emulsification by bile and enzymatic breakdown by pancreatic lipase occurring simultaneously. Secretin, discovered in 1902 as the first hormone ever identified, is released by S cells in the duodenum in response to acidic chyme. This hormone primarily stimulates the release of bicarbonate-rich pancreatic juice, ensuring that the acidic stomach contents are properly neutralized. Secretin also inhibits gastric acid secretion and slows gastric emptying, providing negative feedback that prevents the duodenum from being overwhelmed by acidic chyme. The pancreatic duct system provides the pathway through which pancreatic juice reaches the duodenum. The main pancreatic duct, also called the duct of Wirsung, extends the entire length of the pancreas, collecting secretions from smaller ducts throughout the organ. This main duct joins with the common bile duct to form the ampulla of Vater, which opens into the duodenum through the major duodenal papilla. This anatomical arrangement allows for the coordinated release of both pancreatic juice and bile, optimizing the conditions for fat digestion. Some individuals also possess an accessory pancreatic duct known as the duct of Santorini, which provides an alternate pathway for pancreatic secretions. This anatomical variation reflects the complex embryological development of the pancreas and can sometimes be clinically significant in cases of pancreatic disease. The endocrine function of the pancreas, while representing only about 2% of the organ's mass, plays a crucial role in coordinating digestion with overall metabolic needs. The islets of Langerhans, scattered throughout the pancreatic tissue like small islands, contain several types of hormone-producing cells. Beta cells produce insulin, which lowers blood glucose levels and promotes nutrient storage. Alpha cells secrete glucagon, which raises blood glucose levels by stimulating glycogen breakdown and glucose production. Delta cells produce somatostatin, which inhibits both insulin and glucagon release, providing fine-tuned control of glucose homeostasis. This endocrine function is intimately connected with the digestive process, as nutrients absorbed from the small intestine directly influence pancreatic hormone release. The insulin released in response to rising blood glucose levels not only promotes glucose uptake by cells, but also stimulates protein synthesis and fat storage, effectively channeling absorbed nutrients into appropriate metabolic pathways. The pancreas also produces several other important substances that contribute to digestive function. Pancreatic polypeptide, released by PP cells in the islets, helps regulate pancreatic secretion and gastric emptying. Various growth factors produced by pancreatic cells help maintain the health of the digestive tract lining and promote healing after injury. The vulnerability of the pancreas to disease reflects its crucial role in digestion and metabolism. Pancreatitis, inflammation of the pancreas, can severely compromise both digestive and endocrine function. Pancreatic cancer, while relatively rare, is particularly dangerous because of the organ's deep location and vital functions. Diabetes mellitus results from inadequate insulin production or function, disrupting the body's ability to properly utilize absorbed nutrients. The pancreas also demonstrates remarkable adaptability in response to different dietary patterns. Individuals who consume high-protein diets show increased production of proteolytic enzymes, while those eating high-fat diets produce more lipase. This adaptive capacity allows the digestive system to optimize its function based on habitual dietary patterns, demonstrating the dynamic nature of digestive physiology. As the chyme continues its journey through the duodenum, it encounters the full force of pancreatic secretion. The acidic mixture from the stomach is quickly neutralized by bicarbonate ions, creating the alkaline environment necessary for optimal enzyme function. Proteins begin their systematic dismantling by pancreatic proteases, fats are attacked by lipases, and complex carbohydrates yield to amylase. This enzymatic assault transforms the partially digested chyme into a mixture of smaller molecules that can be absorbed by the small intestine, setting the stage for the next crucial phase of the digestive journey. In the upper right quadrant of the abdominal cavity, protected by the lower ribs and nestled against the diaphragm, lies the largest internal organ in the human body. The liver, weighing approximately 1.5 kilograms in the average adult, serves as both the body's primary chemical processing plant and its most sophisticated detoxification center. This remarkable organ performs over 500 distinct functions, many of which are essential for life itself. Yet for the journey of digestion we are following, its most visible contribution comes in the form of bile, a complex fluid that transforms the impossible task of digesting fats into an elegant process of emulsification and absorption. The liver's anatomical structure reflects its multifaceted role in human physiology. Divided into two primary lobes, the larger right lobe and the smaller left lobe, the organ also contains two smaller divisions, the quadrate lobe and the caudate lobe. These lobes are further subdivided into functional units called lobules, hexagonal structures that serve as the basic working units of hepatic function. Each lobule contains a central vein surrounded by plates of hepatocytes, the primary liver cells, arranged in a radial pattern like the spokes of a wheel. The blood supply to the liver is unique among human organs, receiving blood from two distinct sources. The hepatic artery delivers oxygenated blood directly from the heart, providing the oxygen necessary for the liver's high metabolic demands. However, approximately 75% of the liver's blood supply comes through the portal vein, which carries nutrient-rich blood directly from the digestive tract. This arrangement allows the liver to process absorbed nutrients before they enter the general circulation, serving as the body's first screening and processing center for everything we consume. Within each lobule, blood flows from the periphery toward the central vein through specialized capillaries called sinusoids. These sinusoids are lined with fenestrated endothelium, creating openings that allow direct contact between the blood and hepatocytes. This intimate relationship enables the liver cells to continuously monitor and modify the composition of blood, removing toxins, storing nutrients, and releasing essential proteins and other substances as needed. The hepatocytes themselves are among the most metabolically active cells in the human body, containing abundant mitochondria, endoplasmic reticulum, and other organelles necessary for their diverse functions. These cells can synthesize proteins, metabolize carbohydrates and fats, store vitamins and minerals, and produce bile acids, all while maintaining their own structural integrity and supporting the liver's remarkable regenerative capacity. Bile production represents one of the liver's most important contributions to digestion. Each day, hepatocytes produce between 600 and 1,000 milliliters of bile, a complex fluid containing bile salts, phospholipids, cholesterol, bilirubin, and various ions dissolved in water. This greenish yellow fluid serves multiple functions in digestion, but its primary role involves the emulsification of dietary fats, transforming large fat droplets into smaller particles that can be efficiently processed by pancreatic enzymes. The bile salts, derived from cholesterol through a complex series of enzymatic reactions, represent the most functionally important components of bile. These amphiphilic molecules possess both hydrophilic and hydrophobic regions, allowing them to act as biological detergents. The primary bile acids, cholic acid and chenodeoxycholic acid, are conjugated with either glycine or taurine to form bile salts that remain soluble in the alkaline environment of the small intestine. The synthesis of bile acids from cholesterol serves dual purposes. It provides the raw materials for fat emulsification while simultaneously serving as a major pathway for cholesterol elimination from the body. This process is so significant that it represents the primary mechanism through which the body regulates cholesterol levels, with increased bile acid synthesis leading to reduced cholesterol stores. Bilirubin, the yellow-brown pigment that gives bile its characteristic color, represents the end product of hemoglobin breakdown. When red blood cells reach the end of their 120-day lifespan, they are broken down by macrophages in the spleen and liver, releasing hemoglobin. The heme portion of hemoglobin is converted to bilirubin, which is then transported to the liver, bound to albumin. Hepatocytes conjugate this unconjugated bilirubin with glucuronic acid, creating conjugated bilirubin that can be safely excreted in bile. The gallbladder, a small pear-shaped sac measuring about 8 centimeters in length, serves as bile's storage and concentration facility. Located on the underside of the liver, this muscular organ can hold approximately 50 milliliters of bile, but its importance far exceeds its modest size. During fasting periods, bile produced by the liver flows into the gallbladder through the cystic duct, where it undergoes significant concentration through water and electrolyte absorption. The gallbladder mucosa contains specialized cells that can absorb up to 90% of the water from stored bile, increasing its concentration of bile salts and other components by as much as tenfold. This concentration process is crucial for efficient fat digestion, as concentrated bile provides a more powerful emulsifying action when released into the duodenum. The release of bile is precisely coordinated with the arrival of fat-containing chyme in the duodenum. As lipids enter the small intestine, specialized cells called I cells release cholecystokinin, CCK, a hormone that causes powerful contractions of the gallbladder smooth muscle. Simultaneously, CCK causes relaxation of the sphincter of Oddi, a muscular valve that controls bile flow into the duodenum. This coordinated response ensures that bile is released exactly when and where it is needed for optimal fat digestion. The emulsification process accomplished by bile salts is a marvel of surface chemistry. Large fat droplets, which might measure several micrometers in diameter, are broken down into millions of smaller droplets measuring less than 1 micrometer across. This dramatic increase in surface area provides exponentially more sites for pancreatic lipase to act upon, transforming fat digestion from a slow, inefficient process into a rapid and nearly complete breakdown of dietary lipids. The bile salts arrange themselves around fat droplets with their hydrophobic tails oriented toward the lipid core and their hydrophilic heads facing outward toward the aqueous environment. This arrangement stabilizes the small droplets and prevents them from coalescing back into larger masses. The result is a stable emulsion that provides optimal conditions for enzymatic fat digestion. Beyond its role in fat emulsification, bile serves several other important functions in the digestive process. It helps neutralize acid chyme entering from the stomach, contributing to the alkaline environment necessary for pancreatic enzyme function. Bile also contains immunoglobulin A, providing antimicrobial protection in the small intestine and various growth factors that help maintain the health of the intestinal lining. The enterohepatic circulation represents one of the most elegant recycling systems in the human body. After bile salts have completed their emulsifying work in the small intestine, approximately 95% are actively reabsorbed in the terminal ileum and return to the liver via the portal circulation. This recycling allows the bile salt pool to circulate 4 to 12 times per day, providing efficient fat digestion while conserving these valuable molecules. The liver's role in nutrient processing extends far beyond bile production. As absorbed nutrients arrive via the portal circulation, hepatocytes process carbohydrates by storing excess glucose as glycogen or converting it to fat for long-term storage. Amino acids are used for protein synthesis, converted to glucose when needed, or processed for waste elimination. Fats are processed into various forms for transport and storage throughout the body. The liver also serves as a crucial storage site for many vitamins and minerals. Fat-soluble vitamins A, D, E, and K are stored in significant quantities, providing reserves that can last for months or even years. The liver stores enough vitamin A to meet the body's needs for up to 2 years, while vitamin B12 stores can last for several years. Iron is stored as ferritin, ready for mobilization when needed for hemoglobin synthesis. Detoxification represents another critical hepatic function that impacts digestive health. The liver's cytochrome P450 enzyme system metabolizes not only drugs and environmental toxins, but also various compounds produced by intestinal bacteria. This detoxification capacity helps protect the body from potentially harmful substances that might be absorbed along with nutrients. The liver's synthetic functions are equally impressive. It produces albumin, the major protein responsible for maintaining blood oncotic pressure and serving as a transport protein for various substances. Clotting factors synthesized by the liver are essential for proper blood coagulation, while complement proteins support immune function. The liver also produces transferrin for iron transport, ceruloplasmin for copper transport, and numerous other proteins essential for normal physiology. Glucose homeostasis represents one of the liver's most vital functions. During fed states, excess glucose is stored as glycogen through the process of glycogenesis. During fasting, the liver releases glucose through glycogenolysis, breaking down stored glycogen to maintain blood glucose levels. When glycogen stores are depleted, the liver can produce glucose from amino acids and other substrates through gluconeogenesis. The liver's remarkable regenerative capacity sets it apart from most other organs. Hepatocytes retain the ability to divide throughout life, and the liver can regenerate up to 75% of its mass if damaged. This regenerative capacity is crucial given the liver's exposure to potentially toxic substances and its high metabolic activity. Bile duct obstruction, whether from gallstones, tumors, or other causes, can have serious consequences for both digestive function and overall health. When bile cannot flow normally into the duodenum, fat digestion becomes severely impaired, leading to steatorrhea, the passage of fatty, foul smelling stools. Fat soluble vitamin deficiencies can develop, and the backup of bile can lead to jaundice and liver damage. Gallstones, formed when bile components crystallize out of solution, represent one of the most common disorders affecting the biliary system. Cholesterol stones, the most common type in developed countries, form when bile becomes supersaturated with cholesterol. Pigment stones, composed primarily of bilirubin, are more common in individuals with hemolytic disorders or liver disease. The composition of bile can be influenced by diet, medications, and various disease states. High-fat diets stimulate increased bile production, while certain medications can alter bile composition or flow. Hormonal changes during pregnancy can affect gallbladder function, leading to increased risk of gallstone formation. As the concentrated bile from the gallbladder mixes with the chyme in the duodenum, it begins its crucial work of fat emulsification. The large fat droplets that survive the stomach's churning are quickly broken down into countless smaller particles, creating the optimal conditions for pancreatic lipase to complete the digestion of dietary fats. This transformation, accomplished through the elegant chemistry of bile salts, represents one of the most important steps in making the nutrients from our food available to our cells. The liver continues its multiple functions as this process unfolds, processing the absorbed nutrients that will soon arrive via the portal circulation, producing the proteins necessary for nutrient transport and maintaining the metabolic balance that allows our cells to thrive. Together with the gallbladder's precisely timed bile release, these organs ensure that the complex process of fat digestion proceeds smoothly, setting the stage for the absorption processes that await in the small intestine. Beyond the pyloric sphincter lies the most remarkable stretch of the digestive journey, a tube so intricately designed for its purpose that it represents one of evolution's greatest achievements in biological engineering. The small intestine, measuring approximately 6 to 7 meters in length when fully extended, coils through the abdominal cavity like a carefully wound rope, its seemingly modest external appearance belying an internal architecture of breathtaking complexity and efficiency. This extraordinary organ is divided into three distinct anatomical and functional regions, each specialized for specific aspects of the digestive and absorptive processes. The duodenum, the shortest section at roughly 25 centimeters, serves as the mixing chamber where chyme from the stomach encounters the powerful secretions from the pancreas and liver. The jejunum, extending for approximately 2.5 meters, represents the primary site of nutrient absorption, where most of the chemical breakdown of food is completed and the majority of nutrients enter the bloodstream. The ileum, the longest section at nearly 3.5 meters, specializes in the absorption of specific nutrients like vitamin B12 and bile salts, while also serving as the final checkpoint before materials pass into the large intestine. What makes the small intestine truly extraordinary is not just its length, but the magnificent amplification of its surface area through a series of increasingly intricate structural modifications. If the small intestine were simply a smooth tube, its absorptive surface would measure only about 0.3 square meters, insufficient to handle the complex task of nutrient absorption. Instead, through an elegant hierarchy of folds, projections, and microscopic extensions, the actual absorptive surface area reaches an astounding 200 square meters, roughly the size of a tennis court, all contained within the confines of the abdominal cavity. The first level of this surface amplification comes from the pliques circulars, also known as the valves of Kirkring. These permanent circular folds of the intestinal wall extend partially around the inner circumference of the small intestine, creating a spiral pattern that increases surface area by approximately threefold. More importantly, these folds serve to slow the passage of chyme, ensuring adequate mixing with digestive juices and providing sufficient time for absorption to occur. This rapid turnover ensures that the absorptive surface remains in optimal condition despite constant exposure to digestive enzymes, mechanical abrasion, and potential toxins. The energy cost of this cellular renewal is substantial, with the small intestine consuming about 20% of the body's total energy expenditure during rest, reflecting the metabolic demands of maintaining this crucial interface between the external environment and internal circulation. The duodenum, as the first section of the small intestine, bears the responsibility of neutralizing the acidic chyme arriving from the stomach and coordinating the release of digestive secretions. The duodenal mucosa contains specialized cells that monitor the chemical composition of the arriving chyme and respond by releasing appropriate hormones. Secretin is released in response to acid, stimulating bicarbonate secretion from the pancreas. Cholecystokinin, CCK, is released in response to fats and proteins, triggering the release of pancreatic enzymes and bile from the gallbladder. The Brunner's glands, found only in the duodenum, produce an alkaline, mucus-rich secretion that provides additional buffering capacity and protection for the duodenal mucosa. These glands are most numerous near the pyloric junction, where the need for acid neutralization is greatest. Their secretions help create the optimal pH environment for pancreatic enzyme activity while protecting the intestinal wall from acid damage. As chyme moves into the jejunum, the focus shifts from acid neutralization to nutrient processing and absorption. The jejunum is characterized by tall, densely packed villi and numerous plicae circulares, reflecting its primary role in absorption. The jejunal mucosa produces various enzymes and transport proteins optimized for processing the major nutrients, carbohydrates, proteins, and fats. Carbohydrate absorption in the jejunum involves a series of precisely coordinated steps. Complex carbohydrates that survived gastric processing are further broken down by pancreatic amylase into smaller oligosaccharides and disaccharides. The brush border enzymes then complete the process, with lactase splitting lactose into glucose and galactose, sucrase cleaving sucrose into glucose and fructose, and maltase breaking maltose into two glucose molecules. These monosaccharides are then transported across the enterocyte membrane through specific transport proteins. Glucose and galactose absorption occurs through a sodium-dependent transport process that couples the movement of these sugars to the movement of sodium ions. This active transport system can concentrate glucose in the enterocyte to levels higher than in the intestinal lumen, ensuring efficient absorption even when glucose concentrations are low. Fructose, by contrast, is absorbed through facilitated diffusion via specific fructose transporters. Protein absorption follows an equally sophisticated pathway. The pancreatic proteases break down proteins into smaller peptides and some amino acids, but the majority of protein absorption actually occurs in the form of D and tripeptides rather than individual amino acids. The brush border peptidases complete the final stages of protein digestion, while specialized transport systems move both individual amino acids and small peptides across the intestinal epithelium. The absorption of peptides is particularly interesting from an evolutionary perspective, as it provides a more efficient mechanism than amino acid transport alone. Peptide transporters can handle a wider variety of substrates and often have higher transport capacities than individual amino acid transporters. Once inside the enterocyte, most peptides are quickly broken down to amino acids by intracellular peptidases before being released into the portal circulation. Fat absorption represents perhaps the most complex of all nutrient absorption processes, involving not only the intestinal epithelium but also the lymphatic system. The products of fat digestion, fatty acids, monoglycerides, and fat-soluble vitamins, FACE, are incorporated into micelles formed by bile salts. These micelles diffuse to the brush border membrane, where their contents are absorbed by the enterocytes. Inside the enterocyte, fatty acids and monoglycerides are transported to the endoplasmic reticulum, where they are reassembled into triglycerides. These triglycerides, along with fat-soluble vitamins, cholesterol, and phospholipids, are then packaged into large lipoprotein particles called chylomicrons. Unlike water-soluble nutrients that enter the portal circulation directly, chylomicrons are too large to enter blood capillaries and instead enter the lymphatic system through the lacteals. The ileum, the final section of the small intestine, has several specialized functions that are crucial for overall digestive health. Most notably, the ileum is the only site where vitamin B12 can be absorbed, a process that requires the presence of intrinsic factor produced by the stomach's parietal cells. This absorption occurs in the terminal ileum through specific receptors that recognize the B12 intrinsic factor complex. The ileum is also responsible for the reabsorption of bile salts, a process that is essential for the maintenance of the bile salt pool. Approximately 95% of bile salts are actively reabsorbed in the terminal ileum and returned to the liver via the enterohepatic circulation. This recycling process is so efficient that the total bile salt pool circulates multiple times during a single meal, maximizing the utilization of these important molecules. Peyer's patches, collections of lymphoid tissue found primarily in the ileum, represent an important component of the gut-associated lymphoid tissue, GALT. These structures monitor the intestinal contents for potential pathogens and coordinate immune responses when necessary. The presence of substantial immune tissue in the ileum reflects the importance of immune surveillance at this critical junction between the small and large intestines. The motility patterns of the small intestine are carefully coordinated to optimize both mixing and propulsion of contents. During the fed state, segmentation contractions predominate, creating a back and forth mixing motion that enhances contact between chyme and the absorptive surface. Between meals, migrating motor complexes sweep through the small intestine, clearing any remaining debris and preparing for the next meal. The small intestine also produces various hormones that coordinate digestive function and communicate with other organs. Glucagon-like peptide 1, GLP-1, and glucose-dependent insulinotropic polypeptide, GIP, are released in response to nutrients and help regulate blood glucose levels by stimulating insulin release. These incretin hormones represent an important link between nutrient absorption and metabolic regulation. Water and electrolyte absorption in the small intestine is equally impressive, with approximately 8 to 9 liters of fluid absorbed daily. This includes not only water from food and drinks, but also the various digestive secretions, saliva, gastric juice, bile, and pancreatic juice, MRS, that are added to the intestinal contents during the digestive process. The efficient reabsorption of this fluid is essential for maintaining proper hydration and preventing dehydration. Sodium absorption drives much of the water reabsorption in the small intestine through osmotic gradients created by active sodium transport. Chloride follows sodium passively, while potassium and other electrolytes are handled through specialized transport mechanisms. This coordinated absorption of water and electrolytes maintains the proper consistency of the intestinal contents while ensuring that essential minerals are conserved. The regulation of small intestinal function involves complex interactions between neural, hormonal, and local factors. The enteric nervous system, sometimes called the second brain, contains more neurons than the spinal cord and can coordinate many digestive functions independently of central nervous system input. This local control allows the small intestine to respond quickly to changes in the composition and volume of intestinal contents. Hormonal regulation provides longer term coordination between different parts of the digestive system. The incretin hormones GLP-1 and GIP not only regulate insulin release, but also slow gastric emptying and reduce food intake, providing feedback mechanisms that prevent overwhelming of the absorptive capacity. Other hormones like motilin coordinate the migrating motor complexes that clean the small intestine between meals. The small intestine also serves as an important barrier against harmful substances and pathogens. The tight junctions between enterocytes create a selective barrier that allows nutrients to pass while excluding toxins and bacteria. The rapid turnover of the intestinal epithelium helps maintain this barrier function, while the extensive immune system in the intestinal wall provides active protection against potential threats. Disorders of the small intestine can have profound effects on nutritional status and overall health. Celiac disease, an autoimmune condition triggered by gluten, causes inflammation and villus destruction that severely impairs absorption. Crohn's disease can affect any part of the small intestine, causing inflammation that disrupts normal digestive function. Short bowel syndrome, often resulting from surgical removal of portions of the small intestine, can compromise the absorptive capacity so severely that specialized nutritional support becomes necessary. The remarkable efficiency of the small intestine becomes apparent when one considers that under normal circumstances, less than 5% of ingested nutrients are lost in the feces. This extraordinary absorption efficiency is achieved through the elegant coordination of structure and function from the macro-scale folding of the intestinal wall to the molecular level transport processes occurring at the brush border membrane. As the processed chyme nears the end of its journey through the small intestine, it has been transformed from the acidic mixture that left the stomach into a liquid containing primarily water, electrolytes, fiber, and other unabsorbable materials. The vast majority of nutrients, proteins broken down to amino acids, carbohydrates converted to simple sugars, and fats processed into absorbable forms, have crossed the intestinal barrier and entered either the portal circulation bound for the liver or the lymphatic system for eventual delivery to the bloodstream. This transformation represents one of the most crucial processes in human biology, the conversion of external food into internal nourishment. The small intestine, with its remarkable architecture of absorption, serves as the primary gateway through which the outside world becomes part of our internal environment, where nutrients are sorted, processed, and delivered to sustain every cell in the human body. As the remaining intestinal contents approach the ileocecal valve that guards the entrance to the large intestine, they carry with them only what could not be absorbed, setting the stage for the final processing that awaits in the colon. Proteins enter our digestive system as complex, folded molecules that once served as the structural framework, enzymatic machinery, and functional components of other living organisms. These intricate biological sculptures, composed of chains of amino acids linked together like beads on a string, must be systematically dismantled and reconstructed to become part of our own flesh and blood. The process of protein digestion represents one of the most sophisticated recycling programs in nature, where the building blocks of life are carefully deconstructed, sorted, and made available for reassembly into uniquely human forms. The journey of protein digestion begins the moment we take our first bite of protein containing food. Whether it's the muscle fibers of meat, the storage proteins of grains, the structural proteins of vegetables, or the complex proteins found in dairy products, each type presents unique challenges for the digestive system. Some proteins are tightly wound and cross-linked, requiring aggressive treatment to unfold their structures. Others are protected by cellular membranes or bound to other molecules, necessitating preliminary processing before the main digestive enzymes can access them. In the mouth, protein digestion is minimal but not entirely absent. The mechanical action of chewing begins to disrupt the physical structure of protein-containing foods, breaking cell walls and membranes that might otherwise protect proteins from enzymatic attack. The slightly acidic environment of saliva, with its pH of about 6.8, begins to alter protein structures subtly, though no significant enzymatic breakdown occurs at this stage. However, this initial processing is crucial for what follows, as it increases the surface area available for gastric enzymes and begins the process of protein denaturation. The stomach represents the first major site of protein digestion, where the harsh acidic environment and powerful proteolytic enzymes work in concert to begin the systematic breakdown of protein structures. The hydrochloric acid produced by parietal cells serves multiple crucial functions in protein digestion beyond simply providing the proper pH for enzyme activity. The acid environment, with its pH ranging from 1.5 to 3.5, causes protein denaturation, a process that unfolds the complex three-dimensional structures of proteins and exposes their internal amino acid sequences to enzymatic attack. This denaturation process is essential because proteins in their native, folded state often protect their internal amino acid bonds from enzymatic cleavage. The acid environment disrupts the hydrogen bonds, salt bridges, and hydrophobic interactions that maintain protein structure, converting tightly folded proteins into extended, random coils that are much more accessible to digestive enzymes. Additionally, the acid environment helps solubilize proteins that might otherwise remain bound to food particles or cellular structures. Pepsinogen, the inactive precursor of the stomach's primary protein digesting enzyme, is secreted by chief cells located primarily in the gastric glands of the fundus and body. This zymogen activation system represents a crucial safety mechanism that prevents the stomach from digesting itself. Pepsinogen remains harmlessly inactive until it encounters the acidic environment of the gastric lumen, where the low pH triggers a conformational change that converts it to its active form, pepsin. The conversion of pepsinogen to pepsin is both pH dependent and autocatalytic, meaning that once small amounts of pepsin are formed, they accelerate the conversion of remaining pepsinogen molecules. This positive feedback mechanism ensures rapid enzyme activation when acidic conditions are established, but also means that pepsin activity can be quickly terminated when acid production stops and gastric pH rises. Pepsin itself is a remarkably hardy enzyme, optimally active at pH 1.5 to 2, conditions that would destroy most other proteins. This extreme acid tolerance allows pepsin to function in the hostile environment of the stomach, while other enzymes would be denatured and inactivated. Pepsin belongs to a class of enzymes called endopeptidases, which means it cleaves proteins at internal sites rather than removing amino acids from the ends of protein chains. The specificity of pepsin reflects its role as the initiator of protein digestion. Rather than completely breaking down proteins to amino acids, pepsin's function is to create initial breaks in protein chains, converting large proteins into smaller polypeptides that can be more easily handled by other digestive enzymes. Pepsin preferentially cleaves peptide bonds adjacent to aromatic amino acids like phenylalanine, tyrosine, and tryptophan, as well as leucine and methionine. This specificity pattern results in the production of polypeptides of varying lengths, typically containing 8 to 15 amino acids. The mechanical action of the stomach works synergistically with chemical digestion to maximize protein breakdown. The powerful contractions of the gastric antrum create a grinding action that physically disrupts protein-containing food particles while simultaneously mixing them with gastric juice. This mechanical processing is particularly important for tough, fibrous proteins like those found in meat, where physical breakdown is necessary to expose internal protein structures to enzymatic attack. As the partially digested proteins move from the stomach into the duodenum, they encounter a dramatically different environment. The acidic chyme is quickly neutralized by bicarbonate from the pancreas, raising the pH to approximately 8 to 8.5, an alkaline environment that inactivates pepsin but creates optimal conditions for pancreatic proteases. This pH transition marks a crucial shift in the protein digestion process, from the initial breakdown accomplished by pepsin to the more comprehensive digestion carried out by pancreatic enzymes. The pancreatic proteases represent one of the most sophisticated enzyme systems in human biochemistry, capable of systematically dismantling proteins into their component amino acids. Like pepsin, these enzymes are secreted as inactive precursors to prevent self-digestion of the pancreas. The activation cascade begins with trypsinogen, which is converted to trypsin by enterokinase, an enzyme produced by the duodenal mucosa. Once activated, trypsin serves as the master activator, converting other pancreatic enzyme precursors to their active forms. Trypsin, once activated, becomes a powerful endopeptidase with specificity complementary to that of pepsin. While pepsin preferentially cleaves proteins near aromatic amino acids, trypsin targets peptide bonds adjacent to the basic amino acids lysine and arginine. This complementary specificity ensures that proteins receive comprehensive enzymatic treatment, with different enzymes attacking different sites within the same protein molecules. Chymotrypsin, activated from chymotrypsinogen by trypsin, adds another dimension to pancreatic protein digestion. This enzyme preferentially cleaves peptide bonds adjacent to large aromatic amino acids, including phenylalanine, tryptophan, tyrosine, and leucine. The combined action of trypsin and chymotrypsin, with their overlapping but distinct specificities, ensures that most peptide bonds in dietary proteins are susceptible to enzymatic cleavage. Elastase, another pancreatic endopeptidase, specializes in breaking down elastin and other fibrous proteins that give structure to connective tissues. This enzyme is particularly important for digesting the tough, structural proteins found in meat, where elastin fibers can otherwise resist digestion by other proteases. Elastase preferentially cleaves peptide bonds adjacent to small, uncharged amino acids like alanine and valine. The exopeptidases carboxypeptidase A and carboxypeptidase B work from the ends of peptide chains, removing amino acids one at a time from the carboxyl terminus. Carboxypeptidase A removes aromatic and branched chain amino acids, while carboxypeptidase B removes basic amino acids. These enzymes work in concert with the endopeptidases to ensure complete protein breakdown, attacking peptides from both internal sites and chain termini. The products of pancreatic protein digestion are primarily oligopeptides containing 2 to 8 amino acids, along with some free amino acids. This mixture then encounters the brush border of the small intestine, where the final stages of protein digestion occur. The brush border contains numerous peptidases that complete the breakdown of oligopeptides to amino acids and small peptides that can be absorbed. The brush border peptidases represent a diverse group of enzymes with overlapping specificities that ensure comprehensive peptide digestion. Aminopeptidases remove amino acids from the amino terminus of peptides, while tripeptidases and dipeptidases cleave 3 and 2 amino acid peptides, respectively. The combined action of these enzymes reduces most dietary proteins to amino acids and small peptides suitable for absorption. The absorption of protein digestion products occurs through sophisticated transport systems in the small intestinal epithelium. Contrary to early assumptions that protein absorption occurred solely as individual amino acids, research has revealed that a significant portion of protein-derived nutrients is absorbed as dipeptides and tripeptides through specific peptide transporters. This peptide transport system often has higher capacity and efficiency than individual amino acid transporters, representing an evolutionary optimization of protein absorption. The PEPT1 transporter, located on the apical membrane of intestinal enterocytes, can transport a wide variety of dipeptides and tripeptides into the cell. This transporter is coupled to hydrogen ion movement, taking advantage of the acidic microenvironment at the brush border to drive peptide uptake. Once inside the enterocyte, most peptides are rapidly hydrolyzed to amino acids by intracellular peptidases before being transported across the basolateral membrane into the portal circulation. Amino acid absorption occurs through multiple transport systems, each specialized for different classes of amino acids. The neutral amino acid transporter handles amino acids like alanine, serine, and threonine, while the basic amino acid transporter manages lysine, arginine, and histidine. Acidic amino acids are absorbed through their own specialized transport system, and aromatic amino acids have yet another dedicated transporter. This diversity of transport systems ensures efficient absorption of all amino acids while preventing competition between different amino acid types. The regulation of protein digestion involves complex interactions between neural, hormonal, and local factors. The sight, smell, or thought of protein rich food can trigger the cephalic phase of gastric secretion, increasing both acid and pepsinogen production in preparation for protein digestion. The presence of protein in the stomach stimulates gastrin release, which further enhances gastric secretion. In the small intestine, cholecystokinin, CCK, is released in response to amino acids and peptides, stimulating pancreatic enzyme secretion and coordinating the overall digestive response. The fate of absorbed amino acids depends on the body's immediate needs and overall nutritional status. Some amino acids are used immediately for protein synthesis, replacing worn out proteins in muscles, organs, and other tissues. Others may be converted to glucose through gluconeogenesis when carbohydrate stores are low, or used as precursors for important biological molecules like neurotransmitters, hormones, and nucleic acids. The efficiency of protein digestion is remarkable under normal circumstances, with healthy individuals absorbing 99% of dietary protein. This high efficiency reflects the comprehensive nature of the protein digestion system, with multiple enzymes working in sequence to ensure complete breakdown of even the most complex protein structures. The redundancy built into the system, with multiple enzymes having overlapping specificities, provides backup mechanisms that maintain protein digestion even when individual components are compromised. Protein quality, determined by amino acid composition and digestibility, varies significantly among different protein sources. Animal proteins generally contain all essential amino acids in proportions similar to human needs and are highly digestible, earning them the designation of complete proteins. Plant proteins, while often lacking one or more essential amino acids, can provide complete nutrition when consumed in appropriate combinations. The digestibility of plant proteins is sometimes lower due to the presence of anti-nutritional factors like protease inhibitors and lectins, but proper food preparation can largely overcome these limitations. Certain individuals may have difficulty digesting specific proteins due to genetic variations or acquired conditions. Lactose intolerance affects the ability to digest milk proteins indirectly by preventing proper milk digestion, while celiac disease represents an immune response to gluten proteins that damages the small intestinal lining. Food allergies involve immune responses to specific proteins, while protein energy malnutrition can impair the production of digestive enzymes necessary for proper protein digestion. The recycling aspect of protein digestion extends beyond dietary proteins to include the body's own proteins. Approximately 70 grams of endogenous proteins are secreted into the digestive tract daily in the form of digestive enzymes, mucus, sloughed intestinal cells, and other secretions. These endogenous proteins are digested and reabsorbed along with dietary proteins, representing an important conservation mechanism that prevents the loss of valuable amino acids. As the products of protein digestion are absorbed in the small intestine and enter the portal circulation bound for the liver, they carry with them the potential to become part of every tissue in the human body. The amino acids derived from the muscle of animals or the storage proteins of plants will be reassembled according to human genetic instructions, becoming human muscle, human enzymes, human hormones, and all the other proteins that make up our physical being. This transformation from foreign protein to human protein represents one of the most fundamental processes of life where the building blocks of other organisms are reconstructed to sustain our own existence. Carbohydrates enter our digestive system as nature's primary energy currency, ranging from the simple sweetness of fruit sugars to the complex architectural frameworks of plant starches and the tough structural polymers of dietary fiber. These carbon based molecules, built from basic units of sugar, linked together in chains of varying length and complexity, represent the most readily available fuel for human cellular metabolism. The process of carbohydrate digestion transforms these diverse molecular structures into the simple sugars that can power every process in the human body, from the electrical activity of neurons to the mechanical work of muscle contraction. The diversity of dietary carbohydrates presents unique challenges for the digestive system. Simple sugars like glucose, fructose, and galactose can be absorbed directly without further processing, while disaccharides such as sucrose, lactose, and maltose require enzymatic cleavage to split them into their component monosaccharides. Complex carbohydrates like starch must undergo extensive breakdown to liberate the glucose units locked within their branched and linear structures. Meanwhile, structural carbohydrates, like cellulose and other forms of dietary fiber, resist human digestive enzymes entirely, serving different but equally important roles in digestive health. The journey of carbohydrate digestion begins in the mouth, with the action of salivary amylase, also known as ptyalin. This enzyme represents the first chemical assault on complex carbohydrates, beginning the process of starch breakdown even as food is being mechanically processed by chewing. Salivary amylase is an endoamylase, meaning it attacks starch molecules at random internal sites rather than removing glucose units from the ends of chains. This random cleavage pattern rapidly reduces the size of starch molecules, converting them to shorter chains called dextrins and some maltose molecules. The effectiveness of salivary amylase depends heavily on the duration of contact with food, which varies significantly based on eating habits and food texture. Thorough chewing not only increases the physical surface area available for enzyme action, but also prolongs the time during which salivary amylase can work. Foods that require extensive chewing, such as whole grains and raw vegetables, may undergo substantial starch digestion in the mouth, while rapidly consumed foods or liquids receive minimal salivary processing. The slightly acidic environment of mixed saliva, with its pH of approximately 6.8, provides optimal conditions for amylase activity while beginning the process of food lubrication necessary for swallowing. The enzyme continues to function as food is formed into a bolus and prepared for transport to the stomach, though its activity begins to decline as the pH drops due to bacterial acid production and other factors in the oral cavity. The transition from mouth to stomach marks a dramatic shift in the environment for carbohydrate digestion. The highly acidic conditions of the gastric juice, with pH values ranging from 1.5 to 3.5, quickly inactivate salivary amylase, effectively halting enzymatic carbohydrate digestion. However, this acidic interlude serves important functions in carbohydrate processing, helping to solubilize some complex carbohydrates and beginning the breakdown of cellular structures that may protect starch granules from enzymatic attack. In the stomach's acidic environment, some acid hydrolysis of carbohydrates occurs, though this non-enzymatic process is relatively slow and limited compared to the enzymatic digestion that occurs elsewhere. The mechanical churning action of the stomach continues to break down food particles, further exposing starch granules and other carbohydrates to the digestive processes that await in the small intestine. The gastric phase of digestion also regulates the rate at which carbohydrates enter the small intestine, with the pyloric sphincter controlling the flow of chyme to prevent overwhelming of the intestinal processing capacity. The neutralization of acidic chyme as it enters the duodenum creates the alkaline environment necessary for pancreatic amylase activity. This enzyme, secreted by the pancreatic acinar cells, represents the major starch digesting enzyme in human physiology and is responsible for completing the breakdown of complex carbohydrates that began in the mouth. Pancreatic amylase is structurally similar to salivary amylase, but is produced in much larger quantities and works in the optimal environment of the alkalinized small intestine. The activity of pancreatic amylase is remarkable in both its efficiency and specificity. This enzyme can completely digest starch to maltose and other small oligosaccharides within minutes of contact, provided that adequate mixing occurs. The enzyme attacks the alpha 1 4 glycosidic bonds that link glucose units in the linear portions of starch molecules, but cannot cleave the alpha 1 6 bonds found at branch points in amylopectin, the branched form of starch. This limitation means that pancreatic amylase produces not only maltose, but also larger oligosaccharides called limit dextrins that contain branch points. The products of pancreatic amylase digestion create a mixture of maltose, maltotriose, and alpha limit dextrins that must undergo further processing before they can be absorbed. This is where the brush border enzymes of the small intestine play their crucial role in completing carbohydrate digestion. These membrane-bound enzymes are strategically located at the site of absorption, ensuring that the final steps of digestion occur precisely where the resulting monosaccharides can be immediately transported into the enterocytes. The brush border disaccaridases represent one of the most elegant enzyme systems in human biochemistry, with each enzyme specifically adapted to handle particular carbohydrate substrates. Maltase, actually a complex of several related enzymes called maltase glucoamylase, cleaves maltose into two glucose molecules and can also process maltotriose and some of the alpha limit dextrins produced by amylase. This enzyme complex ensures efficient processing of the major products of starch digestion. Sucrase isomaltase represents a fascinating example of enzyme evolution, functioning as a single protein with two distinct catalytic sites. The sucrase portion cleaves sucrose, table sugar, into glucose and fructose, while the isomaltase portion can break down some of the branched oligosaccharides that resist other enzymes. This dual functionality allows efficient processing of both dietary sucrose and the complex products of starch digestion that contain branch points. Lactase occupies a unique position among the brush border enzymes due to its developmental regulation and genetic variability among human populations. This enzyme cleaves lactose, the primary sugar in milk, into glucose and galactose. In most mammals, lactase activity declines after weaning, but many human populations have evolved lactase persistence, maintaining high enzyme activity into adulthood. This evolutionary adaptation reflects the cultural adoption of dairy farming and the nutritional advantages of being able to digest milk throughout life. The absorption of monosaccharides represents the culmination of carbohydrate digestion, where the products of enzymatic breakdown finally enter the body's internal circulation. This process involves sophisticated transport systems that have evolved to handle the different chemical properties of various sugars. The absorption mechanisms reflect both the importance of efficient carbohydrate utilization and the need to prevent the loss of these valuable energy sources. Glucose absorption occurs primarily through the sodium glucose cotransporter 1-SGLT1 located on the apical membrane of intestinal enterocytes. This remarkable transport protein couples the movement of glucose to the movement of sodium ions, taking advantage of the sodium gradient maintained by the sodium-potassium pump to drive glucose uptake even against concentration gradients. This active transport mechanism allows the small intestine to absorb glucose efficiently even when luminal glucose concentrations are relatively low. The sodium glucose cotransporter can concentrate glucose within the enterocyte to levels several times higher than in the intestinal lumen, ensuring virtually complete absorption of dietary glucose under normal circumstances. The transport process is saturable, meaning that extremely high glucose loads can overwhelm the transport capacity, but under normal dietary conditions, glucose absorption approaches 100% efficiency. Galactose, produced by lactose digestion, shares the same transport system as glucose, being absorbed through SGLT1 with equal efficiency. This shared transport mechanism reflects the similar chemical structure of these two sugars and ensures that both components of lactose are efficiently absorbed when lactase successfully cleaves this milk sugar. Fructose absorption follows a different pathway, utilizing the glucose transporter 5-GLUT5, located on the apical membrane, and glucose transporter 2-GLUT2 on the basolateral membrane. Unlike glucose and galactose absorption, fructose transport is not coupled to sodium movement and occurs through facilitated diffusion. This transport mechanism is generally slower than glucose absorption and can become saturated at high fructose loads, sometimes leading to incomplete absorption and osmotic effects in the intestine. The capacity for fructose absorption shows considerable individual variation and can be influenced by dietary adaptation. Regular consumption of fructose-containing foods appears to upregulate fructose transport capacity, while individuals with limited fructose intake may experience digestive discomfort when consuming large amounts of fruit or foods containing high fructose corn syrup. Once absorbed, monosaccharides enter the portal circulation and travel directly to the liver, where they undergo initial metabolic processing before entering the general circulation. The liver serves as a crucial regulatory hub for carbohydrate metabolism, removing excess glucose from the blood when concentrations are high and releasing stored glucose when concentrations fall. This hepatic processing ensures that blood glucose levels remain within the narrow range necessary for optimal brain function and cellular metabolism. The regulation of carbohydrate digestion involves complex interactions between neural, hormonal, and local factors that coordinate enzyme secretion with carbohydrate intake. The sight, smell, or taste of sweet foods can trigger anticipatory responses that prepare the digestive system for carbohydrate processing. The presence of carbohydrates in the small intestine stimulates the release of incretin hormones like GLP-1 and GIP, which not only enhance insulin secretion but also coordinate digestive function with metabolic needs. The incretin response to carbohydrates represents an important link between nutrient absorption and glucose homeostasis. These hormones are released in proportion to the rate and extent of glucose absorption, providing a sophisticated feedback system that matches insulin release to actual nutrient availability. This system helps prevent both hypoglycemia from excessive insulin release and hyperglycemia from inadequate insulin response. Dietary fiber, while not digested by human enzymes, plays crucial roles in carbohydrate metabolism and overall digestive health. Soluble fibers can slow the rate of carbohydrate digestion and absorption, leading to more gradual increases in blood glucose levels and improved glucose tolerance. This effect is particularly important for individuals with diabetes or prediabetes, as it helps prevent rapid spikes in blood sugar that can be difficult to manage. The fermentation of resistant starch and other indigestible carbohydrates by colonic bacteria produces short chain fatty acids that serve as important energy sources for colonocytes and can influence glucose metabolism throughout the entire body. This microbial processing of carbohydrates represents an additional pathway through which dietary carbohydrates contribute to human nutrition beyond their direct digestion and absorption in the small intestine. Individual variations in carbohydrate digestion and tolerance reflect both genetic differences and environmental factors. Lactase persistence varies dramatically among different populations, with Northern Europeans showing the highest rates of adult lactase activity, while many Asian and African populations show age-related decline in lactase production. These variations reflect evolutionary adaptations to different dietary patterns and demonstrate the dynamic relationship between human genetics and nutritional practices. Similarly, the capacity to digest complex carbohydrates can be influenced by habituation and enzyme adaptation. Individuals consuming high-starch diets often show enhanced amylase production, while those following low-carbohydrate diets may experience temporary digestive discomfort when carbohydrate intake is suddenly increased. These adaptations highlight the remarkable plasticity of the digestive system and its ability to optimize function based on dietary patterns. The timing and composition of carbohydrate intake can significantly influence digestive efficiency and metabolic outcomes. Consuming carbohydrates as part of mixed meals generally results in slower digestion and more stable blood glucose responses compared to consuming isolated sugars. The presence of proteins, fats, and fiber can modulate carbohydrate digestion through various mechanisms, including delayed gastric emptying and reduced enzymatic access to starch molecules. Modern food processing has created new challenges for carbohydrate digestion, with highly refined carbohydrates being digested and absorbed much more rapidly than their whole food counterparts. This rapid absorption can lead to steep increases in blood glucose and insulin levels, potentially contributing to insulin resistance and other metabolic problems over time. Understanding these effects has led to renewed interest in food processing methods that slow carbohydrate digestion and promote more gradual nutrient absorption. As the products of carbohydrate digestion enter the bloodstream and begin their distribution throughout the body, they carry with them the potential energy that will fuel cellular processes in every organ system. The glucose molecules that once formed part of a grain of wheat or an apple will soon power the contraction of heart muscle, the synthesis of proteins in the liver, and the electrical activity of brain neurons. This transformation from plant energy storage to human cellular fuel represents one of the fundamental processes that connect us to the broader web of life, where solar energy, captured by photosynthesis, becomes the chemical energy that sustains human existence. The digestion of fats presents the digestive system with its most formidable challenge, a problem that arises from the fundamental incompatibility between lipids and water. While proteins and carbohydrates dissolve readily in the aqueous environment of the digestive tract, fats stubbornly separate, forming large droplets that resist the water-based enzymatic systems responsible for their breakdown. Yet the human body has evolved an elegant solution to this challenge, transforming what could be an insurmountable obstacle into a precisely orchestrated process of emulsification, digestion, and absorption that rivals any chemical engineering system. Dietary fats arrive in the digestive system in numerous forms, each presenting unique challenges for processing. Triglycerides, the most abundant dietary lipids, consist of three fatty acid chains attached to a glycerol backbone, creating molecules that are entirely hydrophobic. Phospholipids, major components of cell membranes, combine fatty acid chains with phosphate-containing head groups, creating molecules with both hydrophobic and hydrophilic regions. Cholesterol and cholesterol esters add another layer of complexity, while fat-soluble vitamins hitchhike along with these larger lipid molecules, requiring specialized handling to ensure their absorption. The journey of fat digestion begins modestly in the mouth and stomach, where mechanical processing and some preliminary chemical changes prepare lipids for the major digestive processes that await in the small intestine. In the mouth, chewing disrupts the cellular structures that contain fats, releasing them from their biological packaging and increasing the surface area available for enzymatic attack. The warm environment of the oral cavity also helps melt solid fats, converting them to more fluid forms that can be more easily processed. Lingual lipase, secreted by glands at the base of the tongue, begins the enzymatic attack on dietary fats. This enzyme, which remains active in the acidic environment of the stomach, can hydrolyze up to 30% of dietary triglycerides, primarily targeting the outer fatty acid chains. While this may seem modest compared to the comprehensive fat digestion that occurs later, this early lipase activity serves important functions, creating products that stimulate hormonal responses and beginning the process of fat breakdown before the major digestive machinery is engaged. The stomach continues this preliminary processing through both mechanical and chemical means. The powerful churning action of the gastric antrum creates a crude emulsion of fat droplets suspended in the aqueous chyme, though this emulsion is unstable and temporary. Gastric lipase, secreted by chief cells in the gastric fundus, joins lingual lipase in beginning triglyceride hydrolysis. These gastric lipases are particularly important for milk fat digestion in infants, where they can account for a significant portion of total fat digestion. The acidic environment of the stomach also begins to alter the physical properties of dietary fats, helping to solubilize some components while initiating the breakdown of complex lipid structures. However, the major transformation of fat digestion occurs when the lipid-containing chyme enters the duodenum and encounters the sophisticated emulsification system that awaits there. The entry of fat-containing chyme into the duodenum triggers a cascade of hormonal responses that coordinate the release of the substances necessary for efficient fat digestion. Cholecystokinin, CCK, released by eye cells in the duodenal mucosa in response to fatty acids and amino acids, serves as the master coordinator of fat digestion. This hormone stimulates the powerful contraction of the gallbladder, forcing concentrated bile into the common bile duct, while simultaneously causing relaxation of the sphincter of Oddi, allowing bile to flow into the duodenum precisely when it is needed. Bile represents one of evolution's most ingenious solutions to the problem of fat digestion. This complex fluid, produced continuously by hepatocytes and concentrated in the gallbladder, contains bile salts that function as biological detergents capable of transforming large, hydrophobic fat droplets into stable emulsions of microscopic particles suspended in an aqueous medium. The amphiphilic nature of bile salts, with their hydrophilic head groups and hydrophobic steroid backbones, allows them to orient at the interface between lipids and water, dramatically reducing surface tension and preventing the coalescence of fat droplets. The emulsification process begins immediately upon the mixing of bile with fat-containing chyme in the duodenum. Large fat droplets, which may measure several micrometers in diameter, are rapidly broken down into millions of smaller droplets measuring less than 1 micrometer across. This transformation increases the total surface area of the fat by several orders of magnitude, converting a small amount of surface available for enzymatic attack into an enormous interface where pancreatic lipase can work efficiently. Pancreatic lipase, the primary fat digesting enzyme in human physiology, faces the unique challenge of working at the interface between lipid and aqueous phases. This enzyme cannot simply dissolve in the aqueous phase and encounter its substrates randomly, as is the case with proteases and carbohydrases. Instead, pancreatic lipase must bind to the surface of fat droplets and work at the lipid-water interface, a process that requires specialized cofactors and optimal conditions for efficient function. Colipase, a small protein cofactor secreted by the pancreas, plays a crucial role in pancreatic lipase function. This protein, initially secreted as procolipase and activated by trypsin, serves as an anchor that helps pancreatic lipase bind to fat droplets in the presence of bile salts. Without colipase, bile salts would actually inhibit pancreatic lipase activity by preventing the enzyme from accessing its lipid substrates. Colipase essentially allows the enzyme to work in the very environment that makes its substrates available for digestion. The action of pancreatic lipase is both efficient and specific. This enzyme preferentially hydrolyzes the outer fatty acid chains from triglyceride molecules, leaving a monoglyceride with a single fatty acid attached to the central carbon of the glycerol backbone. This specificity is important because monoglycerides are excellent emulsifying agents themselves, helping to stabilize the emulsion and facilitate further digestion. The products of lipase action, fatty acids and monoglycerides, must then be solubilized for absorption, a process that requires another level of bile salt function. Phospholipase A2, another important pancreatic enzyme, handles the digestion of phospholipids, the complex lipids that are major components of cell membranes. This enzyme specifically cleaves the fatty acid from the second carbon of phospholipids, producing lysophospholipids and free fatty acids. The lysophospholipids are themselves potent emulsifying agents and contribute to the overall efficiency of fat digestion and absorption. Cholesterol esterase, also secreted by the pancreas, breaks down cholesterol esters to free cholesterol and fatty acids. This enzyme is important for the absorption of cholesterol from both dietary sources and biliary cholesterol that is recycled through the enterohepatic circulation. The free cholesterol produced by this enzyme, along with cholesterol that was already in free form, is incorporated into micelles for absorption. The absorption of fat digestion products represents one of the most complex processes in human physiology, involving multiple transport mechanisms and cellular processing steps that differ fundamentally from the absorption of water soluble nutrients. As my cells approach the brush border of intestinal enterocytes, their contents are absorbed through a process that is not fully understood but appears to involve both passive diffusion and protein mediated transport. The uptake of fatty acids appears to involve specific fatty acid transport proteins, including fatty acid transport protein 4-FATP4 and the fatty acid translocase CD36. These proteins facilitate the movement of fatty acids across the apical membrane of enterocytes, though simple diffusion may also contribute to fatty acid uptake, particularly for shorter chain fatty acids. Monoglycerides are absorbed through mechanisms that are less well characterized but appear to involve both protein mediated transport and passive diffusion. The formation of chylomicrons occurs in the endoplasmic reticulum and is refined in the Golgi apparatus through a complex process that requires the synthesis of apolipoprotein B48, the major structural protein of chylomicrons. This process is so important that individuals with genetic defects in chylomicron formation develop severe fat malabsorption and deficiencies of fat-soluble vitamins, demonstrating the crucial role of this packaging system in fat absorption. Unlike water-soluble nutrients that enter the portal circulation directly, chylomicrons are too large to enter the blood capillaries and instead enter the lymphatic system through the lacteals, the lymphatic capillaries located in the center of each intestinal villus. This lymphatic route allows chylomicrons to bypass the liver initially and enter the general circulation through the thoracic duct, which empties into the subclavian vein near the heart. The lymphatic absorption of fat serves several important functions beyond simply accommodating the large size of chylomicrons. It allows for the absorption of fat-soluble vitamins, which are incorporated into chylomicrons and transported along with the fats. It also provides a mechanism for the absorption of other lipophilic substances, including some drugs and environmental contaminants that may be present in food. Essential fatty acids, those that cannot be synthesized by the human body and must be obtained from the diet, are absorbed through the same mechanisms as other fatty acids. Linoleic acid and alpha-linolenic acid, the parent compounds of the omega-6 and omega-3 fatty acid families respectively, are incorporated into chylomicrons and distributed throughout the body, where they serve crucial structural and signaling functions. The efficiency of essential fatty acid absorption is generally high, but deficiencies can occur in conditions that impair fat digestion and absorption. The absorption of fat-soluble vitamins A, D, E, and K depends entirely on the efficient digestion and absorption of fats. These vitamins are incorporated into my cells along with other lipid digestion products and absorbed by the enterocytes through mechanisms that are still being elucidated. Some fat-soluble vitamins require specific transport proteins or binding proteins for efficient absorption, while others appear to be absorbed through more general lipid transport mechanisms. Vitamin A absorption is particularly complex, involving the conversion of dietary beta-carotene to retinol in the intestinal mucosa, followed by esterification and incorporation into chylomicrons. Vitamin D absorption appears to be relatively simple, with the vitamin being incorporated into chylomicrons along with other lipids. Vitamin E exists in multiple forms in the diet, with alpha tocopherol being the most biologically active and efficiently absorbed form. Vitamin K absorption involves both the natural forms found in plants and the synthetic forms used in supplements and fortified foods. The regulation of fat digestion and absorption involves multiple feedback mechanisms that coordinate the process with the body's needs and the rate of fat intake. The release of CCK in response to fat in the duodenum not only stimulates bile and pancreatic enzyme release, but also slows gastric emptying, ensuring that fats are delivered to the small intestine at a rate that can be efficiently processed. This hormonal regulation helps prevent the overwhelming of the digestive capacity and ensures optimal fat absorption. Individual variations in fat digestion capacity can have significant clinical implications. Some people produce insufficient pancreatic lipase due to pancreatic disease, leading to steatorrhea and malabsorption of fats and fat-soluble vitamins. Others may have impaired bile acid synthesis or transport, resulting in similar problems with fat digestion. These conditions require dietary modifications and often enzyme replacement therapy to maintain adequate nutrition. The efficiency of fat absorption is normally very high, with healthy individuals absorbing 95–99% of dietary fats. This remarkable efficiency reflects the sophisticated nature of the fat digestion and absorption system, with its multiple redundant mechanisms and precise regulation. However, this efficiency can be compromised by various factors, including certain medications, intestinal diseases, and genetic disorders affecting lipid metabolism. As the absorbed lipids complete their transformation into chylomicrons and enter the lymphatic circulation, they carry with them not only the energy stored in fatty acids, but also the essential building blocks for cell membranes, signaling molecules, and fat-soluble vitamins. The fats that once formed part of a plant seed or animal tissue have been broken down, processed, and repackaged in a uniquely human form, ready to contribute to the maintenance and function of every cell in the body. This remarkable transformation from dietary lipid to human lipid represents one of the most sophisticated aspects of human digestion, where the challenge of processing hydrophobic nutrients in an aqueous environment has been solved through an elegant combination of emulsification, enzymatic processing, and specialized absorption mechanisms that ensure these crucial nutrients reach their cellular destinations. Within the grand symphony of digestion, vitamins and minerals perform as the subtle but essential orchestral elements that enable the entire performance to proceed. These micronutrients, required in amounts measured in milligrams or even micrograms rather than grams, serve as cofactors, catalysts, and structural components that make possible the countless biochemical reactions occurring throughout the body. Their absorption represents one of the most elegant aspects of digestive physiology, where specialized transport systems, regulatory mechanisms, and storage facilities work in concert to ensure that these precious substances are captured from the diet and distributed to the tissues that require them. The fundamental distinction between fat-soluble and water-soluble vitamins creates two entirely different absorption pathways, each adapted to the unique chemical properties of its respective vitamin group. This division reflects not merely academic classification, but represents a profound difference in how these essential nutrients are processed, transported, stored, and utilized within the human body. Understanding this distinction provides insight into why vitamin deficiencies develop differently, why some vitamins can accumulate to toxic levels while others are rapidly excreted, and why the timing and composition of meals can significantly influence micronutrient status. Water soluble vitamins, including the 8B vitamins and vitamin C, share the common characteristic of dissolving readily in aqueous solutions. This solubility allows them to be absorbed directly into the portal circulation and transported immediately to the liver and other tissues. However, their water solubility also means that they are readily excreted in urine when present in excess, providing a natural protection against toxicity, but also requiring regular replenishment from dietary sources. The absorption of water-soluble vitamins occurs primarily in the jejunum through mechanisms that range from simple diffusion to highly sophisticated, actively regulated transport systems. Vitamin C absorption exemplifies the complexity that can be involved in what might seem like straightforward nutrient uptake. This vitamin is absorbed through sodium-dependent vitamin C transporters, SVCT1 and SVCT2, that actively concentrate the vitamin in enterocytes and other tissues. The transport is saturable, meaning that absorption efficiency decreases as intake increases, explaining why massive doses of vitamin C supplements are largely wasted. The B vitamins present a fascinating array of absorption mechanisms, each reflecting the unique chemical properties and biological importance of individual vitamins. Thiamine B1 is absorbed through both passive diffusion at high concentrations and active transport at physiological concentrations. The active transport system involves thiamine transporters that are regulated by thiamine status, increasing in number when thiamine intake is low and decreasing when intake is adequate. This adaptive response helps maintain thiamine homeostasis despite variations in dietary intake. Riboflavin B2 absorption occurs through specific riboflavin carriers that transport the vitamin across the intestinal epithelium. These transporters are pH dependent and work most efficiently in the slightly acidic environment of the proximal small intestine. The absorption process is saturable and regulated by riboflavin status, with efficiency increasing during deficiency states and decreasing when body stores are adequate. Niacin B3 presents an interesting case because it can be absorbed in multiple forms. Nicotinic acid is absorbed through monocarboxylate transporters, while nicotinamide utilizes different transport mechanisms. Additionally, the body can synthesize niacin from the amino acid tryptophan, creating a backup system that can help prevent deficiency when dietary niacin intake is marginal but protein intake is adequate. Pantothenic acid B5 absorption occurs through the sodium-dependent multivitamin transporter SMVT, which also handles biotin transport. This shared transport system creates the potential for competition between these vitamins when intake of one is extremely high, though this is rarely a practical concern with normal dietary patterns. Pyridoxine B6 absorption involves multiple transport mechanisms depending on the chemical form of the vitamin. The most common dietary forms are pyridoxal, pyridoxine, and pyridoxamine, each of which may utilize different transport systems. Once absorbed, all forms are converted to pyridoxal phosphate, the active coenzyme form, primarily in the liver. Biotin absorption occurs through the same sodium-dependent multivitamin transporter that handles pantothenic acid. Biotin is unique among the B vitamins because significant amounts are synthesized by intestinal bacteria particularly in the colon. While the contribution of bacterial biotin synthesis to human nutrition remains debated, it likely provides some protection against biotin deficiency in healthy individuals. Folate absorption represents one of the most complex vitamin absorption processes, involving multiple steps that convert dietary folates to the absorbable form. Most dietary folate exists as polyglutamates with multiple glutamic acid residues attached to the folate molecule. These must be cleaved by brush border folate conjugase, also called pteryl polyglutamate hydrolase, to produce monoglutamate folate, the form that can be absorbed. This hydrolysis step can be inhibited by various factors, including zinc deficiency and certain medications, potentially leading to folate malabsorption. Vitamin B12 absorption stands as perhaps the most complex and highly regulated vitamin absorption process in human physiology, reflecting both the vitamin's crucial importance and its scarcity in natural food sources. This elaborate system begins in the stomach, where B12 must be liberated from food proteins through the action of gastric acid and pepsin. Free B12 then binds to haptocorin, also called R protein, a glycoprotein secreted in saliva and gastric juice that protects B12 from degradation in the acidic stomach environment. The B12 haptocorin complex travels to the duodenum, where pancreatic proteases cleave haptocorin, releasing B12 to bind with intrinsic factor, a glycoprotein synthesized and secreted by gastric parietal cells. The formation of the B12 intrinsic factor complex is essential for absorption, as B12 cannot be absorbed in significant amounts without intrinsic factor under normal physiological conditions. The B12 intrinsic factor complex travels through the small intestine until it reaches the terminal ileum, the only site where B12 absorption can occur. Here, specific receptors called cubum receptors recognize and bind the complex, initiating endocytosis. Once inside the ileal enterocytes, B12 is separated from intrinsic factor and bound to transcobalamin 2, the transport protein that carries B12 in the plasma to tissues throughout the body. This elaborate absorption mechanism explains why B12 deficiency can develop in several different ways: inadequate dietary intake, primarily in strict vegetarians; insufficient gastric acid production, common in elderly individuals; lack of intrinsic factor production, pernicious anemia; or damage to the terminal ileum, Crohn's disease, or surgical resection. The complexity also explains why B12 deficiency develops slowly, as the liver stores large amounts of B12 that can last for several years, even when absorption is completely blocked. Fat-soluble vitamins A, D, E, and K follow the same general absorption pathway as dietary fats, being incorporated into micelles and absorbed along with other lipids. However, each fat-soluble vitamin has unique aspects to its absorption, transport, and metabolism that reflect its specific biological functions and chemical properties. Vitamin A absorption is complicated by the fact that this vitamin exists in multiple forms in the diet. Preformed vitamin A, retinol, and retinol esters is found in animal products, while provitamin A, carotenoids, primarily beta-carotene, are found in plant foods. Both forms are incorporated into micelles for absorption, but they follow different metabolic pathways once absorbed. Retinol esters are hydrolyzed by pancreatic and brush border enzymes to release retinol, which is then absorbed by enterocytes and reesterified for incorporation into chylomicrons. Beta carotene and other provitamin A carotenoids are cleaved by beta carotene oxygenase enzymes in the intestinal mucosa to produce retinol, though this conversion is relatively inefficient and highly variable among individuals. The absorbed vitamin A is transported to the liver in chylomicrons, where it is stored as retinyl esters in hepatic stellate cells. The liver contains enough vitamin A to meet the body's needs for months or even years, which explains why vitamin A deficiency is primarily a problem in areas with chronic dietary inadequacy rather than temporary shortfalls. Vitamin D absorption is relatively straightforward compared to other fat-soluble vitamins, with both vitamin D, 2-ergocalciferol, and vitamin D, 3-cholecalciferol, being incorporated into micelles and absorbed passively. However, the biological activity of absorbed vitamin D depends on subsequent hydroxylation reactions in the liver and kidneys that convert it to the active hormone calcitriol. The efficiency of vitamin D absorption is generally high, but can be impaired by fat malabsorption disorders. Additionally, vitamin D synthesis in the skin through sun exposure can provide a significant portion of the body's vitamin D needs, making dietary absorption less critical for vitamin D status in individuals with adequate sun exposure. Vitamin E absorption involves the uptake of multiple tocopherol and tocotrienol compounds, with alpha tocopherol being the most biologically active form. All forms of vitamin E are absorbed through the same mechanisms as other fat-soluble vitamins, but the body selectively retains alpha tocopherol through the action of alpha tocopherol transfer protein in the liver. Iron absorption exemplifies the sophisticated regulation that characterizes mineral homeostasis. Iron exists in two main forms in food: heme iron from animal sources and non-heme iron from plant sources and iron-fortified foods. These forms are absorbed through completely different mechanisms and with vastly different efficiencies. Heme iron absorption occurs through a specific heme transporter, HCP1, located on the apical membrane of duodenal enterocytes. This transport system is relatively efficient and is not significantly affected by other dietary components, explaining why heme iron has much higher bioavailability than non-heme iron. Once inside the enterocyte, heme is cleaved by heme oxygenase to release iron, which then enters the same intracellular iron pools as non-heme iron. Non-heme iron absorption is far more complex and highly regulated. Iron must first be reduced from the ferric Fe3+ to the ferrous Fe2+, formed by duodenal cytochrome B reductase, before it can be transported across the apical membrane by divalent metal transporter 1, DMT 1. This reduction step can be enhanced by reducing agents like vitamin C and inhibited by compounds that bind iron and prevent its reduction. Once inside the enterocyte, iron can either be stored as ferritin or transported across the basolateral membrane into the circulation. The decision between storage and transport is regulated by iron status through a sophisticated molecular system involving iron response elements and iron regulatory proteins. When body iron stores are adequate, more iron is stored as ferritin in the enterocytes and lost when these cells are sloughed off. When iron stores are low, more iron is transported into circulation through ferroportin, the only known iron export protein. The regulation of iron absorption also involves hepcidin, a hormone produced by the liver that serves as the master regulator of systemic iron homeostasis. Hepcidin binds to ferroportin and causes its internalization and degradation, effectively blocking iron absorption when iron stores are adequate or when infection is present. This system prevents iron overload while ensuring adequate iron availability for essential functions. Calcium absorption involves multiple transport mechanisms that operate in different segments of the small intestine and are regulated by vitamin D status and calcium needs. In the duodenum and proximal jejunum, calcium is actively transported through a vitamin dependent pathway involving calcium channels, calcium binding proteins, and calcium pumps. This active transport system is highly regulated and increases dramatically during periods of high calcium need, such as growth, pregnancy, and lactation. In the ileum and colon, calcium absorption occurs primarily through passive paracellular transport that is proportional to calcium concentration and is less tightly regulated. This passive pathway becomes quantitatively important when calcium intake is high, though its efficiency is much lower than active transport. The regulation of calcium absorption involves complex interactions between parathyroid hormone, vitamin D, and calcium sensing mechanisms that adjust absorption based on calcium status and needs. Vitamin D increases calcium absorption by stimulating the synthesis of calcium transport proteins, while parathyroid hormone indirectly enhances absorption by stimulating vitamin D activation. Zinc absorption occurs primarily in the jejunum through mechanisms that are still being elucidated. Multiple transporters appear to be involved, including ZIP4, SLC, 39, A4, and DMT1, which also transports iron. Zinc absorption is influenced by dietary factors, with phytates and fiber reducing absorption, while amino acids and certain organic acids enhance it. The regulation of zinc absorption involves both transcriptional and post-translational mechanisms that adjust transporter activity based on zinc status. Metallothionein, a zinc-binding protein found in enterocytes, serves as a buffer that can either facilitate zinc transport when status is low or sequester zinc when status is adequate. Other trace elements, including copper, manganese, selenium, and others, each have their own specific transport mechanisms and regulatory systems. Many of these systems show remarkable similarity in their basic design: specific transporters for uptake, intracellular binding proteins for buffering and transport, and regulatory mechanisms that adjust absorption based on nutritional status. The liver plays a crucial role in vitamin and mineral metabolism, serving as both a processing center and a storage depot for many micronutrients. Fat-soluble vitamins are stored in significant quantities in the liver, with vitamin A stores capable of lasting for years, and vitamin D stores providing some buffering capacity against seasonal variations in synthesis. Water-soluble vitamins are generally not stored in large amounts, though the liver does contain some reserves of folate and vitamin B12. The liver also serves as the site where many vitamins are converted to their active forms. Vitamin D undergoes its first hydroxylation in the liver, folate is converted to various coenzyme forms, and vitamin A is processed and distributed to tissues throughout the body. These hepatic functions make the liver essential not just for vitamin storage, but for vitamin activation and distribution. As the products of micronutrient absorption enter the circulation and begin their distribution throughout the body, they carry with them the potential to enable countless biochemical reactions: the iron that will help carry oxygen in hemoglobin, the calcium that will trigger muscle contractions, the B vitamins that will facilitate energy metabolism, and the antioxidant vitamins that will protect cellular structures from damage, all represent the successful capture and processing of these precious dietary components. This intricate dance of absorption, regulation, and distribution ensures that every cell in the body has access to the micronutrients it needs to function optimally, transforming simple dietary components into the essential cofactors that make life itself possible. The small intestine performs a delicate balancing act that represents one of the most remarkable achievements in biological engineering. This 20-foot expanse of tissue must simultaneously serve as an efficient absorptive surface for nutrients while functioning as an impermeable barrier against the countless potential threats that accompany our food. The intestinal barrier stands as the guardian between the external world of ingested materials and the internal sanctity of our bloodstream, making split-second decisions about what should be welcomed into the body and what must be excluded. This selective permeability is achieved through an intricate system of physical, chemical, and immunological barriers that work in seamless coordination. The intestinal barrier is not a simple wall, but rather a dynamic, intelligent interface that can distinguish between beneficial nutrients and harmful substances, between helpful bacteria and dangerous pathogens, between self and non self. Understanding this system reveals how the digestive tract serves not only as a processing facility for food, but as one of the body's most important immune organs. The physical barrier begins with the intestinal epithelium, a single layer of cells that separates the intestinal lumen from the underlying tissues. This seemingly fragile barrier, measuring only about 20 micrometers in thickness, must maintain its integrity while allowing the passage of nutrients, water, and other beneficial substances. The epithelial cells, or enterocytes, are bound together by sophisticated protein complexes that create selective gates between cells. Tight junctions represent the most critical component of the physical barrier. These protein complexes form continuous belts around each cell, creating seals that prevent the uncontrolled passage of materials between cells. The tight junction proteins, including claudins, occludins, and junctional adhesion molecules, form selective pores that can open and close in response to various signals. This dynamic regulation allows the intestine to adjust its permeability based on physiological needs and potential threats. The structure of tight junctions varies along the length of the intestine, reflecting the different functions of each region. In the duodenum and jejunum, where most nutrient absorption occurs, tight junctions are relatively permeable to allow efficient nutrient transport. In the ileum and colon, tight junctions are tighter, providing enhanced barrier function as materials progress toward elimination. This gradient of permeability ensures optimal function while maintaining appropriate security. Adherens junctions and desmosomes provide additional structural integrity to the epithelial barrier. These protein complexes create strong mechanical connections between cells, preventing the physical disruption of the epithelial layer during normal digestive processes. The constant churning of intestinal contents, the passage of food particles, and the mechanical stress of digestion all require robust intercellular connections to maintain barrier integrity. The mucus layer represents another crucial component of the intestinal barrier system. This protective coating, secreted by goblet cells scattered throughout the intestinal epithelium, creates a physical and chemical barrier that prevents direct contact between intestinal contents and the epithelial surface. The mucus is composed primarily of mucins, large glycoproteins that form gel-like structures when hydrated. Antimicrobial peptides, secreted by intestinal epithelial cells, add another layer of chemical defense. These small proteins, including defensins, lysozyme, and lactoferrin, have broad spectrum antimicrobial activity against bacteria, fungi, and viruses. Paneth cells, specialized epithelial cells located in the crypts of the small intestine, are particularly rich sources of antimicrobial peptides and serve as guardians of the stem cell compartments. The secretion of antimicrobial peptides is regulated by both constitutive and inducible mechanisms. Some peptides are produced continuously to maintain baseline protection, while others are rapidly upregulated in response to bacterial products or inflammatory signals. This dual approach provides both preventive protection and rapid response capability. Secretory immunoglobulin, a SIGA, represents the most abundant antibody in the intestinal tract and serves as a crucial component of the adaptive immune barrier. This specialized antibody is produced by plasma cells in the intestinal lamina propria and is actively transported across the epithelium by the polymeric immunoglobulin receptor. Once in the intestinal lumen, SIGA binds to bacteria, viruses, and other antigens, preventing their adhesion to the epithelial surface and facilitating their elimination. The production of SIGA is influenced by the local microbial environment, with beneficial bacteria promoting SIGA production, while pathogenic organisms may attempt to evade or degrade these antibodies. The diversity of SIGA antibodies reflects the complex antigenic environment of the intestine, with each individual developing a unique repertoire of intestinal antibodies based on their microbial exposures and dietary patterns. The gut-associated lymphoid tissue, GALT, represents the largest immune organ in the human body, containing more immune cells than the bone marrow and spleen combined. This extensive immune system is strategically positioned throughout the intestinal tract to monitor incoming materials and coordinate appropriate immune responses. The GALT includes both organized lymphoid structures and diffusely distributed immune cells. Peyer's patches are the most prominent organized lymphoid structures in the small intestine, appearing as dome-like collections of immune cells that protrude into the intestinal lumen. These structures are covered by a specialized epithelium that includes microfold M cells, which actively sample antigens from the intestinal contents and transport them to underlying immune cells. This sampling process allows the immune system to continuously monitor the intestinal environment and develop appropriate responses to potential threats. The lamina propria, the connective tissue layer beneath the intestinal epithelium, contains a diverse collection of immune cells, including T cells, B cells, macrophages, dendritic cells, and plasma cells. These cells work together to maintain immune surveillance while preventing inappropriate inflammatory responses to harmless food antigens and beneficial bacteria. The balance between tolerance and immunity in this environment is crucial for intestinal health. Intraepithelial lymphocytes represent a unique population of immune cells that reside between the epithelial cells themselves. These cells, primarily T cells, provide immediate immune surveillance at the epithelial surface and can rapidly respond to threats that breach the initial barrier defenses. Their position allows them to monitor both the luminal environment and the epithelial cells themselves for signs of infection or damage. The regulation of intestinal barrier function involves complex interactions between epithelial cells, immune cells, and the local microbial environment. Inflammatory cytokines can increase barrier permeability by disrupting tight junctions, while anti inflammatory signals help maintain barrier integrity. This dynamic regulation allows the barrier to respond appropriately to different challenges while maintaining its essential protective function. Barrier dysfunction, often referred to as leaky gut syndrome in popular literature, can occur when the normal regulatory mechanisms are disrupted. Various factors, including stress, certain medications, alcohol, infectious agents, and inflammatory conditions, can compromise barrier function. When the barrier becomes excessively permeable, normally excluded substances may enter the systemic circulation, potentially triggering inflammatory responses and contributing to various health problems. The relationship between barrier function and immune tolerance is particularly important in the development of food allergies and autoimmune diseases. The intestinal immune system must learn to tolerate harmless food proteins while maintaining the ability to respond to genuine threats. When this balance is disrupted, the immune system may develop inappropriate responses to food antigens, leading to food allergies or intolerances. Oral tolerance, the process by which the immune system learns not to respond to ingested antigens, is a crucial function of the intestinal barrier system. This process involves specialized dendritic cells that sample antigens and present them to T cells in a tolerogenic manner, promoting the development of regulatory T cells that suppress inflammatory responses. The failure of oral tolerance mechanisms can lead to food allergies and other immune mediated disorders. The regenerative capacity of the intestinal epithelium is crucial for maintaining barrier function despite constant exposure to potentially damaging substances. The entire intestinal lining is renewed every 3 to 5 days through the proliferation of stem cells located in the crypts of Leber-Kuhn. This rapid turnover ensures that damaged cells are quickly replaced and that barrier function is maintained even under challenging conditions. Aging affects multiple aspects of intestinal barrier function, including changes in tight junction proteins, reduced mucus production, altered immune responses, and slower epithelial regeneration. These age-related changes may contribute to increased susceptibility to infections and inflammatory conditions in elderly individuals. Understanding these changes may help develop strategies to maintain barrier function throughout the lifespan. Nutritional factors play important roles in supporting barrier function. Certain amino acids, particularly glutamine, serve as preferred fuel sources for intestinal epithelial cells and help maintain barrier integrity. Omega-3 fatty acids can reduce inflammation and support barrier function, while certain vitamins and minerals serve as cofactors for proteins involved in tight junction formation and maintenance. The intestinal barrier represents one of the most sophisticated interfaces in the human body, where the external environment meets our internal physiology. Its ability to selectively allow beneficial substances to pass while excluding potential threats requires constant vigilance and precise regulation. As nutrients complete their absorption and enter the circulation, they do so under the careful supervision of this remarkable barrier system, which ensures that only appropriate materials gain access to our internal environment. The coordination between barrier function and absorption demonstrates the elegant integration of digestive physiology, where protection and nourishment are achieved simultaneously through complementary mechanisms. This integration becomes even more remarkable when we consider the role of the intestinal microbiome, the vast community of bacteria that calls our digestive tract home and plays crucial roles in both barrier function and overall digestive health. The relationship between our cells, our food, and our microbial partners represents one of the most complex and important aspects of digestive physiology, where the boundaries between self and non self become beautifully blurred in service of our overall health and survival. Within the warm, moist chambers of our digestive tract lives a hidden universe of extraordinary complexity and profound importance. This microscopic ecosystem, known as the gut microbiome, contains trillions of bacterial cells representing hundreds of different species, creating a community so vast and diverse that it rivals any ecosystem found in nature. These microscopic inhabitants are not merely passive passengers in our digestive journey, but active partners in the process of transforming food into nourishment, synthesizing essential compounds, protecting against pathogens, and maintaining the delicate balance that defines digestive health. The numbers alone are staggering. The human gut harbors approximately 100 trillion bacterial cells, a population that actually outnumbers our own human cells. This bacterial community weighs between 1 and 3 kilograms, making it comparable in mass to our liver or brain. Yet despite their abundance, these microorganisms remained largely invisible and unknown until recent advances in molecular biology revealed their presence and began to illuminate their profound influence on human health and digestion. The composition of the gut microbiome is as individual as a fingerprint, shaped by genetics, birth method, early feeding patterns, geographic location, diet, medications, stress, and countless other factors that vary from person to person. While certain bacterial groups are found in most healthy individuals, the specific species and their relative abundances create unique microbial signatures that can vary dramatically between individuals and even within the same person over time. Early feeding patterns further shape the developing microbiome. Breast milk contains not only nutrients for the infant, but also beneficial bacteria and prebiotic compounds called human milk oligosaccharides that selectively promote the growth of beneficial species like bifidobacterium. Formula feeding creates a different microbial environment, often leading to greater bacterial diversity at an earlier age, but potentially missing some of the specific benefits associated with breastfeeding. The introduction of solid foods marks a dramatic shift in microbiome development as new nutrient sources become available and the microbial community begins to mature toward an adult-like composition. This transition period, typically occurring between 6 months and 3 years of age, represents a critical window during which the microbiome establishes patterns that may persist throughout life. The adult microbiome is dominated by two major bacterial phyla, Firmicutes and Bacteroidetes, which together typically account for 80–90% of gut bacteria in healthy individuals. Firmicutes include many species that are particularly efficient at extracting energy from dietary carbohydrates, while Bacteroidetes excel at breaking down complex plant polysaccharides and proteins. The ratio between these two groups, known as the Firmicutes divided by Bacteroidetes ratio, has been associated with various aspects of health and disease, including obesity, metabolic syndrome, and inflammatory conditions. Other important bacterial groups include actinobacteria, which includes beneficial species like bifidobacterium that are particularly abundant in healthy infants, and proteobacteria, a diverse phylum that includes both beneficial and potentially harmful species. The presence of smaller populations of other bacterial groups adds to the overall diversity and functional capacity of the microbiome. The functional capabilities of the gut microbiome extend far beyond what human cells can accomplish alone. Bacteria possess enzymatic machinery for breaking down complex carbohydrates that resist human digestive enzymes, including various forms of dietary fiber, resistant starch, and plant polysaccharides. This bacterial fermentation process occurs primarily in the colon and produces short chain fatty acids, gases, and other metabolites that have important physiological effects. Short chain fatty acids, particularly acetate, propionate, and butyrate, represent some of the most important products of bacterial fermentation. These compounds serve multiple functions. They provide energy for colonocytes, the cells lining the colon, help maintain the intestinal barrier, influence immune function, and can even affect metabolism in distant organs, including the liver and brain. Butyrate, in particular, is the preferred energy source for colonocytes and has anti-inflammatory properties that help maintain colonic health. The production of short-chain fatty acids depends on the availability of fermentable substrates, primarily dietary fiber and resistant starch that escape digestion in the small intestine. Different bacterial species specialize in fermenting different types of substrates, creating a complex web of metabolic interactions where the waste products of some bacteria become nutrients for others. This metabolic cross-feeding creates a stable, self-sustaining ecosystem that can efficiently extract energy and nutrients from materials that would otherwise be lost. Bacterial synthesis of vitamins represents another crucial function of the gut microbiome. Several B vitamins, including biotin, folate, and vitamin B12, can be synthesized by intestinal bacteria, potentially contributing to the host's nutritional status. While the quantitative importance of bacterial vitamin synthesis in humans remains a subject of research, it likely provides some protection against deficiency states and may be particularly important during periods of dietary inadequacy. Vitamin K synthesis by bacteria is perhaps the best established example of microbial vitamin production. Bacteria in the colon, particularly species of Bacteroides and Enterococcus, synthesize menaquinones, vitamin K2, which can be absorbed and contribute to the body's vitamin K status. This bacterial vitamin K production is especially important for newborn infants, who are born with sterile guts and low vitamin K stores, making them vulnerable to vitamin K deficiency, bleeding until their microbiomes become established. The gut microbiome also plays crucial roles in immune system development and function. The constant presence of bacterial antigens provides ongoing stimulation that helps train the immune system to distinguish between harmful pathogens and beneficial microorganisms. This immune education is particularly important during early life, when the adaptive immune system is still developing its responses to various antigens. Beneficial bacteria contribute to immune function through multiple mechanisms. They compete with pathogenic organisms for nutrients and adhesion sites, produce antimicrobial compounds that inhibit the growth of harmful bacteria, and stimulate the production of secretory IgA and other components of the mucosal immune system. Some bacterial species can also directly communicate with immune cells through specialized signaling pathways, influencing the balance between inflammatory and anti-inflammatory responses. The concept of colonization resistance illustrates how the established microbiome protects against pathogenic invasions. A healthy, diverse microbiome occupies ecological niches that might otherwise be colonized by harmful organisms, creates an environment that is hostile to pathogens through the production of organic acids and antimicrobial compounds, and stimulates host immune responses that help clear potential threats. This protective function becomes apparent when the normal microbiome is disrupted by antibiotics, leading to increased susceptibility to infections like Clostridioides difficile colitis. The gut brain axis represents one of the most fascinating aspects of microbiome research, revealing bidirectional communication between the gut microbiome and the central nervous system. Bacteria can produce neurotransmitters and other signaling molecules that influence mood, behavior, and cognitive function. The vagus nerve, which connects the gut to the brain, serves as one pathway for this communication, while bacterial metabolites that enter the circulation can directly affect brain function. Different bacterial species can produce various neurotransmitters. Lactobacillus species can produce GABA, Enterococcus and Streptococcus can produce serotonin, and Bacillus can produce dopamine. While the functional significance of bacterial neurotransmitter production is still being investigated, these findings suggest that the microbiome may influence mood and behavior through direct biochemical pathways. The microbiome's influence on drug metabolism adds another layer of complexity to its biological importance. Bacterial enzymes can metabolize medications, either activating prodrugs or inactivating active compounds. This bacterial drug metabolism can significantly affect drug efficacy and toxicity, with important implications for personalized medicine. The composition of an individual's microbiome may help predict their response to certain medications and guide dosing decisions. Dysbiosis, an imbalance in the gut microbiome characterized by reduced diversity or the overgrowth of potentially harmful species, has been associated with numerous health conditions, including inflammatory bowel disease, irritable bowel syndrome, obesity, diabetes, cardiovascular disease, and even certain cancers. While it remains unclear whether dysbiosis is a cause or consequence of these conditions, the associations are strong enough to suggest that maintaining a healthy microbiome is important for overall health. Factors that can disrupt the gut microbiome include antibiotic use, which can dramatically reduce bacterial diversity and allow the overgrowth of antibiotic-resistant species, dietary changes, particularly shifts toward highly processed, low-fiber diets, stress, which can alter gut motility and immune function in ways that affect bacterial communities, and various medications, including proton pump inhibitors, nonsteroidal anti-inflammatory drugs, and certain antidepressants. The resilience of the microbiome varies among individuals and depends on factors including age, overall health status, and the nature and duration of the disrupting influence. Young, healthy individuals often show remarkable recovery of their microbiomes after antibiotic treatment, while elderly individuals or those with underlying health conditions may experience more persistent changes. Prebiotics and probiotics represent two approaches to supporting microbiome health. Prebiotics are non-digestible compounds, primarily certain types of dietary fiber that selectively promote the growth of beneficial bacteria. These compounds pass through the small intestine largely unchanged and serve as fermentable substrates for beneficial bacteria in the colon. Examples include inulin, fructooligosaccharides, and galactoligosaccharides, which are found naturally in foods like garlic, onions, leeks, asparagus, and bananas. Probiotics are live microorganisms that, when consumed in adequate amounts, confer health benefits to the host. These are typically administered as supplements or in fermented foods like yogurt, kefir, sauerkraut, and kimchi. The most commonly used probiotic species include various lactobacillus and bifidobacterium strains, though other species are also being investigated for their potential benefits. The effectiveness of probiotics depends on multiple factors, including the specific strains used, the dose and duration of administration, the health status of the individual, and the composition of their existing microbiome. While some probiotic interventions have shown benefits in clinical trials, the field is still developing an understanding of which probiotics work best for which conditions and in which individuals. Diet represents perhaps the most powerful tool for shaping the gut microbiome. Different dietary patterns promote the growth of different bacterial communities, with high fiber diets generally associated with greater microbial diversity and the production of beneficial metabolites. The Mediterranean diet, rich in fruits, vegetables, whole grains, and olive oil, has been associated with a microbiome profile linked to better health outcomes. Conversely, Western style diets high in processed foods, refined sugars, and saturated fats have been associated with reduced microbial diversity and the growth of bacteria associated with inflammation and metabolic dysfunction. The speed with which dietary changes can alter the microbiome is remarkable, with significant shifts observable within days of changing eating patterns. Fermented foods represent a unique category of dietary interventions that can directly introduce beneficial bacteria while also providing substrates for existing bacterial communities. These foods, produced through bacterial or yeast fermentation, have been consumed by humans for thousands of years and may have contributed to the coevolution of humans and their microbial partners. The future of microbiome research holds tremendous promise for understanding and treating digestive disorders and other health conditions. Fecal microbiota transplantation, while currently used primarily for recurrent C. difficile infections, is being investigated as a treatment for various other conditions. Personalized nutrition based on microbiome composition may allow for more targeted dietary recommendations, while engineered probiotics designed to produce specific therapeutic compounds represent an exciting frontier in biotechnology. As our understanding of the gut microbiome continues to evolve, it becomes increasingly clear that these bacterial partners are not merely inhabitants of our digestive tract, but integral components of our biological systems. They represent a hidden organ that contributes to digestion, immunity, metabolism, and even behavior in ways that are still being discovered. The relationship between humans and their gut bacteria exemplifies the interconnectedness of life, where the boundaries between organism and environment, self and other, become beautifully blurred in service of mutual survival and thriving. The trillions of bacteria that call our digestive tract home remind us that we are not solitary beings, but walking ecosystems, communities of life forms working together to transform the external world into the internal nourishment that sustains us. As the products of bacterial fermentation join the absorbed nutrients in our circulation, they carry with them the biochemical wisdom of millions of years of coevolution, contributing to the complex symphony of molecules that maintain our health and vitality. In this ancient partnership between human and microbe, we find one of the most profound examples of cooperation in nature, where mutual benefit has created systems of extraordinary sophistication and resilience. As the remnants of our meal complete their journey through the small intestine, having surrendered most of their nutritional treasures to the body's eager absorption mechanisms, they approach the final processing station of the digestive tract. The large intestine, also known as the colon, awaits to perform the concluding acts of this remarkable biological drama. Here, in this final segment of the alimentary canal, the last valuable resources are extracted from what remains. Water is reclaimed with remarkable efficiency, and waste is prepared for its eventual elimination from the body. The large intestine presents a stark contrast to the absorptive complexity of the small intestine, where the small intestine is characterized by its extraordinary length and intricate surface amplifications designed for maximum nutrient extraction. The large intestine is shorter, wider, and organized primarily for water absorption and waste processing. Measuring approximately 1.5 meters in length and 6 centimeters in diameter, the colon forms a frame around the small intestine, beginning in the lower right portion of the abdomen and following a predictable anatomical course that reflects its functional priorities. The journey through the large intestine begins at the ileocecal valve, a one-way sphincter that guards the transition between the ileum and the cecum, the first portion of the colon. This valve serves multiple important functions. It prevents the backflow of colonic contents into the small intestine, regulates the flow of material from the ileum, and helps maintain the distinct bacterial environments of the small and large intestines. The valve opens intermittently in response to meals and certain hormonal signals, allowing measured amounts of ileal contents to enter the colon for final processing. The cecum, a pouch-like structure that forms the beginning of the large intestine, serves as a receiving chamber for material arriving from the ileum. Attached to the cecum is the vermiform appendix, a small finger-like projection that has long been dismissed as a vestigial organ with no important function. However, recent research has revealed that the appendix may serve as a reservoir for beneficial bacteria, helping to repopulate the colon with healthy microbes after episodes of diarrheal illness or antibiotic treatment that might disrupt the normal bacterial community. From the cecum, the large intestine continues as the ascending colon, which travels upward along the right side of the abdominal cavity until it reaches the hepatic flexure, where it makes a sharp turn to cross the upper abdomen as the transverse colon. Another sharp turn at the splenic flexure marks the beginning of the descending colon, which travels down the left side of the abdomen before forming the shaped sigmoid colon. This tortuous course through the abdominal cavity maximizes the contact time between the intestinal contents and the absorptive surface, ensuring efficient water recovery. The final portion of the large intestine is the rectum, a straight segment that serves as a temporary storage chamber for feces before elimination. The rectum is equipped with specialized sensory mechanisms that can distinguish between gas, liquid, and solid contents, allowing for the voluntary control of defecation. The anal canal, with its internal and external sphincters, provides the final control mechanisms for waste elimination. The structure of the large intestinal wall reflects its specialized functions. Unlike the small intestine with its elaborate villi and microvilli, the colonic mucosa is relatively flat, with simple tubular glands called crypts of Lieberkühn extending deep into the underlying tissue. These crypts contain the stem cells that continuously regenerate the colonic epithelium, as well as specialized cells that contribute to water absorption and mucus production. The epithelial lining of the large intestine is dominated by colonocytes, cells that are specialized for water and electrolyte absorption. These cells possess transport proteins that can actively pump sodium out of the intestinal lumen, creating osmotic gradients that drive water absorption. The efficiency of this process is remarkable, with the colon typically absorbing 1 to 2 liters of water daily from the approximately 1.5 to 2 liters of fluid that enter from the ileum. Goblet cells are particularly abundant in the large intestine, reflecting the importance of mucus production in this environment. The mucus produced by these cells serves multiple functions. It lubricates the increasingly solid intestinal contents, protects the epithelial surface from mechanical damage, and provides a habitat for beneficial bacteria while excluding potential pathogens from direct contact with the intestinal wall. Water absorption in the large intestine occurs through both passive and active mechanisms. The active transport of sodium creates the driving force for most water absorption, with chloride following passively to maintain electrical neutrality. Potassium secretion also occurs in the colon, and excessive fluid losses through diarrhea can lead to significant potassium depletion. The regulation of these transport processes allows the colon to adjust water absorption based on the consistency of the arriving contents and the body's hydration status. The bacterial fermentation that occurs in the large intestine represents one of the most important aspects of colonic function. The undigested carbohydrates that arrive from the small intestine, including dietary fiber, resistant starch, and various other plant polysaccharides, serve as substrates for the diverse bacterial community that calls the colon home. This fermentation process produces short chain fatty acids, gases, and other metabolites that have important physiological effects. Short chain fatty acids, particularly butyrate, acetate, and propionate, serve as the primary energy source for colonocytes. Butyrate is especially important, providing up to 70% of the energy needs of colonic epithelial cells. This metabolic arrangement creates a mutually beneficial relationship between the host and the bacterial community. Bacteria receive nutrients from undigested carbohydrates, while the host receives energy-rich metabolites that support colonic health. The production of short-chain fatty acids also helps maintain the slightly acidic environment of the colon, which inhibits the growth of potentially pathogenic bacteria while promoting the growth of beneficial species. This acidic environment, with a pH typically ranging from 5.5 to 6.5 inches the cecum and ascending colon, gradually becomes more neutral as contents move toward the rectum. Gas production is an inevitable consequence of bacterial fermentation in the colon. The major gases produced include hydrogen, carbon dioxide, methane, and hydrogen sulfide, along with smaller amounts of other compounds. The composition and quantity of gas production vary significantly among individuals based on their dietary patterns, bacterial communities, and genetic factors that influence gas metabolism. Most intestinal gas is absorbed into the bloodstream and eliminated through the lungs, but a significant portion must be eliminated through the rectum. The average person produces 400 to 1,500 milliliters of gas daily, though this can vary dramatically based on dietary choices and individual bacterial communities. Foods rich in fermentable carbohydrates, such as beans, cabbage, and certain fruits, can significantly increase gas production in susceptible individuals. The motility patterns of the large intestine are quite different from those of the small intestine, reflecting its different functional priorities. Rather than the rapid propulsive waves that characterize small intestinal motility, the colon exhibits slower, more irregular contractions that mix contents and gradually move them toward the rectum. This slower transit allows for maximum water absorption and thorough bacterial fermentation. Segmentation contractions, similar to those in the small intestine but much slower, create a churning action that mixes colonic contents and brings them into contact with the absorptive surface. These contractions also help break up the increasingly solid material and facilitate the formation of feces. The circular muscle contractions that create segmentation occur at intervals of several minutes rather than the seconds that characterize small intestinal motility. Mass movements represent the most powerful propulsive contractions in the colon. These wave like contractions, which occur only a few times per day, can move colonic contents over large distances very quickly. Mass movements are often triggered by meals through the gastrocolic reflex, which explains why many people feel the urge to defecate shortly after eating, particularly after the first meal of the day. The formation of feces represents the culmination of the digestive process, where the remnants of our food are consolidated into a form suitable for elimination. Normal feces consist of approximately 75% water and 25% solid matter, with the solid component including undigested food residue, bacteria, both living and dead, epithelial cells shed from the intestinal lining, and various metabolic waste products. The consistency of feces depends largely on the efficiency of water absorption in the colon and the transit time through the large intestine. Rapid transit, as occurs in diarrheal conditions, results in insufficient water absorption and loose, watery stools. Slow transit, as occurs in constipation, allows for excessive water absorption, resulting in hard, difficult to pass stools. The brown color of normal feces results from the bacterial breakdown of bilirubin, the yellow pigment derived from hemoglobin breakdown. Bacteria in the colon convert bilirubin to urobilinogen and then to stercobilinogen, which is oxidized to stercobilin, the brown pigment that gives feces its characteristic color. Changes in fecal color can sometimes indicate digestive problems or other health issues. The bacterial component of feces is substantial, with bacteria making up 25–30% of the dry weight of normal stools. These bacteria include both the living members of the colonic microbiome and the dead bacteria that are continuously being shed as part of the normal bacterial turnover. The high bacterial content of feces explains why proper hygiene after defecation is so important for preventing the spread of intestinal bacteria to other body sites or to other individuals. The rectum serves as the final staging area for waste elimination, with specialized mechanisms that allow for the voluntary control of defecation. The rectal walls contain stretch receptors that detect the arrival of fecal material and send signals to the brain indicating the need for elimination. The ability to distinguish between gas, liquid, and solid contents allows for selective elimination and helps prevent accidental soiling. The defecation reflex involves both involuntary and voluntary components. When feces enter the rectum, stretch receptors trigger the relaxation of the internal anal sphincter, an involuntary smooth muscle that normally maintains continence. However, the external anal sphincter, composed of voluntary skeletal muscle, remains contracted unless consciously relaxed. This dual sphincter system allows for conscious control over the timing of defecation. Disorders of large intestinal function can significantly impact quality of life and overall health. Constipation, defined as infrequent or difficult bowel movements, can result from slow colonic transit, inadequate fiber intake, insufficient fluid intake, or various medical conditions. Diarrhea, characterized by frequent, loose stools, can result from infections, inflammatory conditions, malabsorption, or other factors that increase intestinal secretions or decrease absorption. Inflammatory bowel diseases, including Crohn's disease and ulcerative colitis, can severely compromise large intestinal function. These conditions involve chronic inflammation of the intestinal wall, leading to symptoms such as bloody diarrhea, abdominal pain, and weight loss. The inflammation can interfere with normal water absorption and alter the bacterial community, perpetuating the cycle of intestinal dysfunction. Irritable bowel syndrome, a functional disorder of the large intestine, affects millions of people worldwide and is characterized by altered bowel habits, abdominal pain, and bloating. While the exact mechanisms underlying this condition are not fully understood, it appears to involve abnormalities in intestinal motility, bacterial communities, and the communication between the gut and brain. Dietary factors significantly influence large intestinal function. Adequate fiber intake is essential for normal colonic function, providing substrates for beneficial bacterial fermentation while adding bulk to stools and promoting regular elimination. Different types of fiber have different effects. Insoluble fiber adds bulk and speeds transit, while soluble fiber is more readily fermented and produces beneficial short-chain fatty acids. The large intestine also plays important roles in drug absorption and metabolism. While most drug absorption occurs in the small intestine, the colon can absorb certain medications, particularly those designed for local action or sustained release. Some drugs are also metabolized by colonic bacteria, which can either activate prodrugs or inactivate active compounds. As the final act of digestion plays out in the large intestine, the remarkable efficiency of the human digestive system becomes apparent. From the complex meal that entered the mouth hours or days earlier, virtually all useful nutrients have been extracted and absorbed. Water has been reclaimed with impressive efficiency, and waste has been prepared for elimination. The transformation from food to feces represents the completion of one of biology's most fundamental processes, where the external environment is processed, sorted, and selectively incorporated into our internal world. The large intestine's role as the final processing station in this journey reminds us that even in elimination, there is elegance and purpose. The careful recovery of water and electrolytes, the beneficial relationships with bacterial partners, and the controlled removal of waste all serve the greater goal of maintaining the internal environment that sustains life. As waste prepared for elimination awaits its final departure from the body, it carries with it only what could not be used, leaving behind in our circulation and tissues all the nutrients that will fuel the countless processes that define human existence. Throughout our journey from mouth to colon, we have witnessed the remarkable precision with which digestive processes unfold, each step perfectly timed and coordinated with the next. This orchestration does not happen by chance, but results from one of the most sophisticated control systems in human biology. The neural and hormonal networks that govern digestion work in seamless harmony, anticipating needs, responding to changes, and maintaining the delicate balance required for optimal nutrient processing and absorption. The control of digestion involves multiple overlapping systems that operate at different timescales and levels of organization. The autonomic nervous system provides rapid, moment-to-moment control, while hormones offer more sustained regulatory influences that can persist for minutes to hours. Local chemical signals fine tune responses based on the immediate environment, and even mechanical forces contribute to the regulatory network through stretch sensitive mechanisms. At the heart of digestive control lies the enteric nervous system, often called the second brain, due to its remarkable independence and complexity. This intrinsic nervous system of the digestive tract contains more neurons than the spinal cord, with an estimated 400 to 600 million nerve cells distributed throughout the gut wall. These neurons are organized into two main networks: the myenteric plexus, which primarily controls muscle function and motility, and the submucosal plexus, which regulates secretion and blood flow. The enteric nervous system possesses an extraordinary degree of autonomy, capable of coordinating complex digestive functions even when disconnected from the central nervous system. This independence allows the digestive tract to continue functioning during sleep, anesthesia, or spinal cord injuries that interrupt communication with the brain. The enteric nervous system can integrate sensory information, process signals, and generate appropriate motor responses entirely within the gut wall. The neurons of the enteric nervous system include sensory neurons that monitor the chemical composition, pH, osmolarity, and mechanical properties of intestinal contents. These intrinsic sensory neurons can detect the presence of nutrients, toxins, pathogens, and other substances, providing the information necessary for appropriate digestive responses. Motor neurons control the smooth muscle contractions that drive peristalsis and mixing movements, while interneurons coordinate complex patterns of activity across different regions of the digestive tract. The neurotransmitters used by enteric neurons reflect the complexity of digestive control. Acetylcholine serves as the primary excitatory neurotransmitter, stimulating both muscle contractions and glandular secretions. Nitric oxide and vasoactive intestinal peptide, VIP, function as inhibitory neurotransmitters, causing smooth muscle relaxation and coordinating the descending inhibition that characterizes peristaltic waves. Serotonin, produced by enterochromaffin cells in the intestinal lining, serves as both a neurotransmitter and a hormone, influencing motility, secretion, and even mood. The sympathetic nervous system generally inhibits digestive function, redirecting energy and blood flow to other organ systems during times of stress or danger. Sympathetic activation decreases gastric acid secretion, reduces intestinal motility, and constricts blood vessels in the digestive tract. This response makes biological sense, as digestion is not a priority when immediate survival is threatened, but chronic sympathetic activation can significantly impair digestive health. The concept of the gut brain axis has gained increasing recognition as research reveals the intimate connections between digestive function and brain activity. The vagus nerve serves as a major highway for bidirectional communication between the gut and brain, carrying not only motor commands from the brain to the gut, but also sensory information from the gut to the brain. This communication pathway helps coordinate digestive function with overall physiological state and can influence mood, stress responses, and even cognitive function. Hormonal control of digestion involves a complex network of signaling molecules that coordinate digestive function with nutritional needs and metabolic status. These hormones are produced by specialized endocrine cells scattered throughout the digestive tract, creating the largest endocrine organ in the human body. The gastrointestinal hormones can be classified into several families based on their chemical structures and evolutionary relationships. Gastrin, produced by G cells in the gastric antrum and duodenum, serves as one of the most important regulators of gastric function. This hormone is released in response to the presence of protein in the stomach and stimulates gastric acid secretion, promotes gastric motility, and supports the growth and maintenance of gastric mucosa. Gastrin secretion is regulated by both neural and chemical factors, with vagal stimulation and the presence of amino acids and peptides serving as major stimuli. Cholecystokinin, CCK, produced by eye cells in the duodenum and jejunum, coordinates the response to fat and protein in the small intestine. This hormone stimulates pancreatic enzyme secretion, causes gallbladder contraction, relaxes the sphincter of Oddi, and slows gastric emptying to allow adequate time for duodenal processing. CCK also acts as a satiety signal, reducing food intake by activating neural pathways that signal fullness to the brain. Secretin, the first hormone ever discovered, is produced by S cells in the duodenum in response to acidic chyme entering from the stomach. This hormone stimulates pancreatic bicarbonate secretion, which neutralizes gastric acid and creates the optimal pH for pancreatic enzyme function. Secretin also inhibits gastric acid secretion and gastric motility, providing negative feedback that prevents the duodenum from being overwhelmed by acidic contents. The incretin hormones, including glucagon-like peptide 1, GLP-1, and glucose-dependent insulinotropic polypeptide, GIP, represent a crucial link between nutrient absorption and metabolic regulation. These hormones are released by L cells and K cells, respectively, in response to nutrients in the small intestine. They stimulate insulin secretion in a glucose-dependent manner, meaning they only promote insulin release when blood glucose levels are elevated, thus helping to prevent hypoglycemia. The incretin hormones also slow gastric emptying, reduce food intake, and may promote the growth and survival of pancreatic beta cells. These multiple effects make the incretin system a major target for diabetes therapeutics, with several medications designed to enhance or mimic incretin action now available for treating type 2 diabetes. Ghrelin, produced primarily by cells in the gastric fundus, serves as a powerful hunger signal that increases before meals and decreases after eating. This hormone stimulates appetite by acting on the hypothalamus, promotes gastric motility, and may influence the reward pathways associated with food consumption. Ghrelin levels are influenced by meal timing, sleep patterns, and metabolic status, helping to coordinate food intake with energy needs. Peptide YPY, produced by L cells in the ileum and colon, acts as a satiety signal that reduces food intake and slows gastric emptying. This hormone is released in response to nutrients in the distal small intestine and colon, providing feedback about the adequacy of nutrient intake and helping to terminate meals when sufficient food has been consumed. The regulation of these hormonal systems involves complex feedback loops that integrate information about nutritional status, metabolic needs, and digestive capacity. The release of digestive hormones is influenced by neural signals, mechanical factors such as gastric distension, and chemical factors, including the presence of specific nutrients and changes in pH or osmolarity. The cephalic phase of digestive regulation demonstrates the remarkable anticipatory capacity of the digestive control system. The sight, smell, taste, or even thought of food can trigger neural and hormonal responses that prepare the digestive tract for incoming nutrients. This preparatory response includes increased salivary flow, gastric acid secretion, pancreatic enzyme production, and changes in intestinal motility and blood flow. The cephalic phase response is mediated primarily through the vagus nerve and involves learned associations between food cues and digestive responses. This conditioning allows the digestive system to optimize its function based on past experience and anticipated needs. The strength of the cephalic response can be influenced by factors such as hunger level, food preferences, and psychological state. The gastric phase of regulation begins when food actually enters the stomach and continues as gastric processing proceeds. Mechanical distension of the stomach stimulates both neural reflexes and hormonal responses that enhance digestive function. The presence of nutrients in the stomach directly stimulates the release of gastrin and other hormones, while triggering neural pathways that coordinate gastric motility and secretion. The intestinal phase represents the most complex level of digestive regulation, as nutrients entering the small intestine trigger a cascade of hormonal and neural responses that coordinate pancreatic secretion, bile release, intestinal motility, and systemic metabolic adjustments. The intestinal phase also provides important feedback signals that regulate gastric emptying and food intake, ensuring that nutrients are processed at a rate that matches the digestive capacity of the small intestine. Circadian rhythms add another layer of complexity to digestive regulation, with many aspects of digestive function showing predictable daily variations. Gastric acid secretion typically peaks in the evening and is lowest in the early morning, while pancreatic enzyme production and intestinal motility also show circadian patterns. These rhythms help optimize digestive function for typical eating patterns while conserving energy during fasting periods. The molecular clocks that control circadian rhythms are present in digestive tissues and can be influenced by meal timing, suggesting that eating patterns can help synchronize digestive rhythms with metabolic needs. Disruption of circadian rhythms, such as occurs with shift work or jet lag, can impair digestive function and contribute to metabolic disorders. Stress represents a major factor that can disrupt digestive regulation through multiple pathways. Acute stress activates the sympathetic nervous system and the hypothalamic pituitary adrenal axis, leading to reduced digestive activity and altered hormone production. Chronic stress can have more persistent effects on digestive function, contributing to conditions such as irritable bowel syndrome, peptic ulcers, and inflammatory bowel disease. The relationship between stress and digestive function is bidirectional, with digestive problems also capable of influencing stress responses and psychological well-being. This gut-brain connection helps explain why digestive disorders often have psychological components and why stress management can be an important part of treating digestive conditions. Age-related changes affect multiple aspects of digestive regulation, with older adults showing altered hormone production, reduced neural function, and modified responses to regulatory signals. These changes can contribute to digestive problems commonly seen in elderly individuals, including reduced appetite, delayed gastric emptying, and increased susceptibility to digestive infections. Medications can significantly interfere with digestive regulation, either as intended therapeutic effects or as unwanted side effects. Proton pump inhibitors alter gastric pH and can affect nutrient absorption, while antibiotics can disrupt the bacterial communities that contribute to digestive regulation. Understanding these interactions is important for optimizing therapeutic outcomes while minimizing adverse effects on digestive function. The integration of neural and hormonal control systems creates a remarkably robust and adaptable regulatory network that can respond to changing conditions while maintaining overall digestive efficiency. This integration allows the digestive system to function effectively across a wide range of dietary patterns, eating schedules, and physiological states. Individual variations in digestive regulation help explain why people can have different responses to the same foods and why digestive disorders can vary so much in their manifestations. Genetic differences in hormone production, receptor sensitivity, and neural function all contribute to these individual variations, suggesting that personalized approaches to digestive health may become increasingly important as our understanding of these systems continues to develop. As we contemplate the orchestra of signals that coordinate digestive function, we gain appreciation for the remarkable sophistication of biological control systems. The seamless integration of neural reflexes, hormonal cascades, local chemical signals, and mechanical responses creates a regulatory network that rivals any human engineered control system in its complexity and reliability. This biological orchestra, playing its intricate symphony throughout every meal and every day, ensures that the fundamental process of transforming food into nourishment proceeds with the precision and coordination necessary to sustain life. As our journey through the digestive system draws to its conclusion, we arrive at a moment of profound reflection on the extraordinary integration that characterizes human digestion. What began as a simple bite of food has become a symphony of molecular transformations, cellular interactions, and systemic responses that exemplify the elegant complexity of biological systems. The digestive process represents far more than the sum of its individual components, emerging as a masterpiece of coordination where every element works in harmony to maintain the delicate balance we call homeostasis. The concept of homeostasis extends far beyond simple regulation to encompass the dynamic equilibrium that characterizes all living systems. In the context of digestion, homeostasis involves the constant adjustment of countless variables to maintain optimal conditions for nutrient processing while preserving the integrity of the tissues involved. This biological balancing act requires continuous monitoring, rapid responses to changing conditions, and the ability to anticipate and prepare for future needs. The integration of digestive processes operates across multiple temporal scales, from the millisecond responses of neural reflexes to the seasonal adaptations that prepare the body for periods of abundance or scarcity. This temporal integration allows the digestive system to function efficiently in both the short term, processing individual meals, and the long term, maintaining nutritional status across days, weeks, and years. The fed state represents one of the most remarkable examples of physiological integration, as the body coordinates responses across multiple organ systems to process incoming nutrients. The sight and smell of food trigger anticipatory responses that prepare the digestive tract for incoming nutrients. The act of eating activates neural pathways that stimulate digestive secretions and motility. The arrival of nutrients in the small intestine triggers hormonal cascades that coordinate pancreatic secretion, bile release, and metabolic adjustments throughout the body. During the fed state, the body shifts into an anabolic mode characterized by nutrient storage and tissue building. Insulin levels rise in response to absorbed glucose, promoting the uptake and storage of nutrients in muscle, liver, and adipose tissue. Protein synthesis increases as absorbed amino acids become available for tissue repair and growth. The liver switches from glucose production to glucose storage, while adipose tissue shifts from fat breakdown to fat synthesis. This coordinated response to feeding demonstrates the remarkable communication network that links the digestive system to every other organ system in the body. Hormones released by the digestive tract influence brain function, affecting mood, alertness, and the desire for additional food. Nutrients absorbed from the intestine modify liver function, influencing the production of proteins, lipids, and other essential compounds. The products of bacterial fermentation in the colon can affect immune function, inflammation, and even cardiovascular health. The fasted state presents an entirely different pattern of integration as the body shifts its priorities from nutrient processing to nutrient mobilization and conservation. As the time since the last meal increases, blood glucose levels begin to decline, triggering hormonal responses that maintain glucose homeostasis. Glucagon levels rise, stimulating the liver to break down glycogen and release stored glucose into the bloodstream. Growth hormone and cortisol levels increase, promoting the breakdown of fat and protein to provide alternative fuel sources. The digestive tract itself undergoes significant changes during fasting periods. Gastric acid secretion decreases to protect the stomach lining in the absence of food. Pancreatic enzyme production is reduced to conserve resources. Intestinal motility shifts to the migrating motor complex pattern that clears debris from the small intestine and prepares it for the next meal. These fasting adaptations demonstrate the digestive system's ability to modify its function based on nutritional status and energy needs. The transition between fed and fasted states illustrates the dynamic nature of digestive homeostasis. These transitions are not abrupt switches, but gradual shifts that involve the coordinated adjustment of numerous physiological parameters. The timing and magnitude of these adjustments reflect the body's remarkable ability to predict and prepare for changing nutritional conditions based on past experience and circadian rhythms. Circadian regulation adds another layer of integration to digestive homeostasis, synchronizing digestive function with the natural rhythms of day and night. The biological clocks that control these rhythms are present throughout the digestive system, from the brain centers that regulate appetite to the bacterial communities in the colon that show their own circadian patterns of activity. The circadian control of digestion helps optimize nutrient processing for typical eating patterns while conserving energy during normal fasting periods. Gastric acid secretion typically peaks in the evening when food intake is often highest, while pancreatic enzyme production and intestinal absorptive capacity show their own circadian variations. These rhythms can be influenced by meal timing, suggesting that eating patterns can help synchronize digestive function with metabolic needs. The disruption of circadian rhythms, such as occurs with shift work, jet lag, or irregular eating patterns, can significantly impact digestive homeostasis. These disruptions can lead to altered hormone production, modified bacterial communities, and increased susceptibility to digestive disorders and metabolic diseases. Understanding these relationships has important implications for optimizing digestive health in our modern world, where irregular schedules are increasingly common. The integration of digestive function with immune homeostasis represents one of the most critical aspects of gastrointestinal health. The digestive tract must maintain a delicate balance between tolerance and immunity, accepting beneficial nutrients and microorganisms while defending against potential threats. This balance is maintained through complex interactions between epithelial barriers, immune cells, and the microbial community. The concept of oral tolerance exemplifies this integration as the immune system learns to accept food proteins as harmless while maintaining the ability to respond to genuine threats. This process involves specialized antigen presenting cells that sample intestinal contents and present antigens to immune cells in a tolerogenic context. The failure of oral tolerance mechanisms can lead to food allergies and autoimmune diseases, demonstrating the importance of proper immune integration. The gut microbiome plays crucial roles in maintaining both digestive and immune homeostasis. The bacterial community helps train the immune system, produces beneficial metabolites, competes with pathogenic organisms, and contributes to the maintenance of epithelial barrier function. The composition and activity of the microbiome are influenced by diet, medications, stress, and other factors, creating a dynamic ecosystem that must be maintained for optimal health. Metabolic integration represents another crucial aspect of digestive homeostasis, as the absorbed products of digestion must be distributed and utilized throughout the body in ways that maintain energy balance and support cellular function. The liver plays a central role in this integration, processing absorbed nutrients and distributing them according to the body's needs. Excess glucose is stored as glycogen or converted to fat. Amino acids are used for protein synthesis or converted to glucose when needed, and fatty acids are packaged for transport to tissues throughout the body. The integration of digestive function with energy homeostasis involves sophisticated feedback mechanisms that adjust food intake based on energy needs and nutritional status. Hormones produced by the digestive tract communicate information about nutrient availability to brain centers that control appetite and food-seeking behavior. Long-term signals from adipose tissue provide information about energy stores, while short-term signals from the stomach and intestines provide information about recent food intake. This integration of energy regulation with digestive function helps maintain body weight and composition within relatively narrow ranges, despite significant variations in food intake and energy expenditure. The disruption of these regulatory mechanisms can lead to obesity, eating disorders, and other conditions that affect both metabolic and digestive health. The adaptive capacity of the digestive system demonstrates remarkable plasticity in response to changing dietary patterns, environmental conditions, and physiological demands. Individuals consuming high-protein diets show increased production of protein-digesting enzymes, while those following high-carbohydrate diets produce more amylase. The bacterial community adapts to dietary changes, with different bacterial species flourishing under different nutritional conditions. These adaptive responses occur at multiple levels, from changes in gene expression that alter enzyme production to modifications in bacterial community composition that optimize fermentation patterns. The time course of these adaptations varies, with some changes occurring within days, while others may take weeks or months to fully develop. This adaptive capacity allows the digestive system to optimize its function for different dietary patterns and environmental conditions. The resilience of digestive homeostasis becomes apparent when we consider the system's ability to maintain function despite various challenges and disruptions. The digestive tract can continue to process nutrients effectively across a wide range of dietary compositions, eating patterns, and physiological states. Multiple redundant mechanisms ensure that critical functions are maintained even when individual components are compromised. This resilience is built into the system at multiple levels. If one digestive enzyme is deficient, others can often compensate partially for its absence. If one absorption pathway is blocked, alternative routes may become more active. If beneficial bacteria are depleted, the remaining species can often expand to fill vacant ecological niches. This biological redundancy reflects the critical importance of nutrient acquisition for survival and the evolutionary pressure to maintain digestive function under challenging conditions. The aging process presents unique challenges to digestive homeostasis as various components of the system gradually decline in function over time. Gastric acid production may decrease, reducing the efficiency of protein digestion and mineral absorption. Pancreatic enzyme production may decline, potentially affecting fat digestion. The intestinal barrier may become more permeable, allowing the passage of substances that would normally be excluded. The bacterial community may become less diverse, potentially affecting immune function and metabolic health. Despite these age-related changes, the digestive system often maintains adequate function throughout most of the human lifespan, testament to the robustness of its design and the effectiveness of its compensatory mechanisms. Understanding these age-related changes can help guide interventions to maintain digestive health in older adults, including dietary modifications, enzyme supplements, and probiotic interventions. The regenerative capacity of digestive tissues represents one of the most remarkable aspects of gastrointestinal homeostasis. The intestinal lining completely renews itself every few days, the gastric mucosa regenerates continuously, and even the liver can regenerate substantial portions of its mass when damaged. This regenerative capacity requires enormous energy expenditure and sophisticated regulatory mechanisms, but allows the digestive system to maintain function despite constant exposure to potentially damaging substances. The stem cell populations that drive this regeneration are carefully regulated by signals that balance proliferation with differentiation, ensuring that new cells are produced at the right rate and develop into the appropriate cell types. Disruption of these regulatory mechanisms can lead to both inadequate regeneration, resulting in tissue damage, and excessive proliferation, potentially leading to cancer. The concept of digestive reserve capacity helps explain how the system can maintain homeostasis despite various stresses and challenges. Under normal conditions, the digestive system operates well below its maximum capacity, producing more enzymes than strictly necessary, maintaining more absorptive surface area than minimally required, and supporting bacterial communities that are more diverse than essential. This excess capacity provides a buffer that allows the system to maintain function even when individual components are compromised. The measurement and assessment of digestive homeostasis in clinical practice involves multiple approaches, from simple observation of symptoms to sophisticated laboratory tests that measure specific aspects of digestive function. Understanding normal variations in digestive function helps distinguish between physiological adaptations and pathological changes, guiding appropriate interventions when homeostatic mechanisms are overwhelmed. The future of digestive health may increasingly involve personalized approaches that take into account individual variations in genetics, microbiome composition, dietary patterns, and environmental exposures. As our understanding of digestive homeostasis continues to evolve, we may develop more targeted interventions that work with the body's natural regulatory mechanisms rather than against them. The integration of digital health technologies with digestive health monitoring may provide new insights into the dynamic nature of digestive homeostasis. Continuous monitoring of various biomarkers could reveal patterns and relationships that are not apparent from static measurements, leading to better understanding of how digestive function varies over time and in response to different influences.